number of people in the webinar on Zoom, but this is also being live streamed on YouTube. So we're kind of waiting for everyone to be able to log in and get in, but we're going to start off this morning with a few introductions of the organizing team for the webinar. And so I'll start off with Franny Brewer. Just a little bit of background. This team has been organizing this CRB symposium for a few months now. There's a lot of interest across the Pacific, and it started off with just this organizing team and some of the work from the Big Island and Invasive Species Council kickstarted. And this team, especially Elliot Parsons, Franny Brewer, and Keith Weiser, have done a lot of work to get this organized. And we hope you enjoyed this this webinar series. But first off, let's introduce Franny Brewer.
Hi, Andrea. Thank you. Yeah, I'm here on the Big Island of Hawaii. I'm manager of Big Island Invasive Species Committee. And as I said yesterday, we just got our first detection of Coconut Rhinoceros Beetle in the last year. So we're in an early detection, rapid response phase of invasion. It's been really great to hear from all the folks who are sharing how we can hopefully not go forward on that invasion curve. And I really appreciate the help that we've gotten from Keith. Keith, you can go from here.
Thanks, Franny. Yeah, I'm Keith Weiser, and I manage the CRB response in Hawaii. The formal organization called CRB Response. Of course, there's multiple organizations involved, and yeah, happy to be a part of this symposium and hope everybody's getting a lot out of it. I guess we'll pass it to you, Elliot.
Thanks, Keith. Thanks, Andrea. Elliot Parsons. I'm a specialist with the Pacific Risk, or the Regional Invasive Species and Climate Change Management Network, and super excited to have you all with us today. It's been an amazing experience planning this over the last few months with this fabulous planning team, and we're excited to help get this information out there to where it's needed. So thank you so much.
All right, and then for myself, I didn't introduce myself earlier. My name is Andrea Blas. I'm based in Guam and work with the Guam Department of Agriculture Biosecurity Division. I am principal investigator for their CRB management team, but also I am consultant with the Micronesia Conservation Trust, where I am working as the regional research coordinator for invasive species and climate change. And I think we have a lot of people joined on right now, but I just want to do a recap of yesterday. So just a few minutes to go over, we had some great talks yesterday. The sessions started off with some background information about CRB and why we need to care about it, the history of it as it's spread across the Pacific from its native range in Southeast Asia. And then we had some great talks about the experiences on Hawaii, and also the potential for RNAi for use as in CRB control. And a lot of talks also about the difference between eradication and management and then containment for the different islands that are experiencing CRB invasions at the moment. But also there were some great talks and a lot of interest about the AI detection using drone imagery and remote surveillance, some of the harmonic and radio technology being used to track CRB and identify breeding sites. And then we also had a few talks about just trapping and tracking the CRB. And we had a great presentation yesterday from an undergraduate student from UH Manoa about the CRB impact and food security on Pacific Islands. Such an impressive talk by, especially as an undergraduate, Andy Ledoux. I wish I had been that focused and on target for myself at that point in my life. But then we also finished off with some information about CRB communications, and we saw the statewide plan for Hawaii and their communications plan for CRB. And then just as an overview for today, we've got some talks about the genetics of CRB, the detection and invasion pathways, how they're getting around from island to island, and work that can be done for containment of the CRB, and some of the biocontrol efforts that are going on in different islands. And then we're going to wrap up this afternoon with a Q&A session where the presenters from today and yesterday, a selection of them will be available for an open question and answer session, as well as some of the questions that have been put in the chat and in the comments from YouTube over today and yesterday. And so I believe, I think we're ready to go. We may be a little ahead of schedule, but our first speaker today is Dr. Mark Arrow. He's with the Pacific Community, or formerly known as the Secretariat of the Pacific Community. And he is the Land Resources Division Pacific Awareness and Response to the Coconut Rhinoceros Beetle Project, or that's the program that he works with. And so if we can get Mark's presentation queued up, Franny. Great. And I will turn this over to Mark.
Good morning, everyone, and thanks, Andrea, for the introduction. Andrea has introduced me. I am Mark Arrow. I'm with the Land Resources Division of the Pacific Community, and I look after the Coconut Rhinoceros Beetle project within the division. So just before I proceed with the presentation this morning, part of the results that I will be sharing with in the presentation will be from a joint project that we are carrying out in Vanuatu. So I just wanted to initially acknowledge a donor partner, which is MFED, and also our collaborating partners, AgResearch and New Zealand, as well as the Ministry of Agriculture and Forestry in Vanuatu. And also I'd like to take the time to thank the organizing committee for inviting me to run through this presentation. So basically for my presentation, I've been asked to just give an overview of the situation within the Pacific, the Pacific region as a whole, and also share some experiences from the management efforts that we are carrying out in Vanuatu. Next slide, please. Okay, Joe. So just as a background, the incursion of CRB within the region is not a new issue. We had the incursion as early as the 1900s, and as indicated also by some of our presenters earlier, the first incursion was in 1909. That was into Samoa through some potted seedlings that were imported in from Sri Lanka, I think Ceylon at that time. That established within Samoa and then spread throughout the other neighboring Pacific islands. And also we had a second sort of invasion or detection that was picked up during the time of the Second World War, and that was in Palau, as well as the Gazil Peninsula area of PNG. So that was the second, the two sort of incursions that we had within the region. Next slide, please. Okay, so this map just basically shows the incursions that we had, and the corresponding is that they were detected from the various Pacific islands. So the first one into Samoa, and then into the neighboring islands, and then the second detection in Palau and the New Britain area of PNG. Next slide, please. So in terms of the early indication. I think this was also said in Aubrey's presentation earlier. But this is 1973 in Fiji, and this was the kind of damage that was detected. But I think the situation improved once the nudibranch was discovered from its native range within the 1970s, and then distributed around the region at that time. Next slide, please. But then I think, the next, the current invasion that we have now is, of course, the population or the haplotype that is resistant to the CRB. There's also, this was highlighted in detail, the initial detection within the region that was in 2007 from around the Tumon Bay area in Guam. But since then, the population, the resistant population has been detected from a couple of, quite a number of the Pacific Island countries and territories now, which we will look at in the next. Sorry, could we go back? Yeah, okay. So the other thing I just wanted to highlight here is the last bullet point. The concept of the resistant to the virus was already highlighted earlier in some of the publications that were from the Philippines, from the Maldives, as well as from Indonesia. So it's not really a new detection that the resistant population had already existed within the native range. So I think, a note to take also here is if we had to look at the natural enemies, then that may be the region that we may need to look at within the native range. Next slide, please. So the recent indication, just a repeat of what was detected from the, what was the situation from the earlier indication, similar damage. This photo is of the from 2022 in Vanuatu, just caused exactly the same kind of damage that was caused by the initial indication of the beetle. Next slide, please. Okay, this timeline was shared by Aubrey earlier, and he had explained the situation, but the information I just wanted to highlight from here is that the earlier indication, which is which is within the two lines, the purple lines. That's the earlier indication. And in terms of I just wanted to highlight in terms of the spread, or the spread of the spread or the invasion within the region. It took a longer time for the beetle to establish. Whereas if we look at the current indication that's between the orange lines, the spread is quite rapid, and the spread and the establishment is quite rapid. So we are looking at a situation where they are spreading much faster than was the situation with the earlier indication, and we are thinking that may be attributed to the more the improvements within with the mode of transport, and then the movements between the islands in terms of the personal or in terms of human movements, as well as equipment. So that could be attributed to that. Next slide, please. Okay, this is this table. I just wanted to highlight in terms of the proximity, I think, of the initial detection within the individual countries that we've that have been picked up, and I think that the last column, which may be of importance to note, so the earlier indication, the actual sites have not been really confirmed. I think basically because they were detected when they had already spread further within the countries of detection. But with the current detections, we are able to pick up where exactly they have been picked up. And they are more, the areas of initial detection are more relating to sort of international movements. So yeah, the Guam one is, of course, highlighted by Aubrey. It's within Wedding Chapel. But the Hawaii, I think that was also highlighted by Keith in his presentation as well. But it's also within the vicinity of the international airport. And then for the RNMI1, I think it's within the Alba area as well. And for PNG, we have both the recent detection of the population that is related to recent population that is resistant to the virus was also picked up from an hotel that's next to the airport. And then Solomon Islands, it was picked up from within an area where the Pacific Festival of Arts was held 2 years earlier. And then Vanuatu, the first, because they have both the virus susceptible as well as the resistant population. The one that's susceptible was picked up from a village, and it was suspected that there were some sand aggregates that were brought in directly around there from an infested country. And also the virus-resistant population has been picked up from around the wharf area, the international wharf. And the New Caledonia is also related. The initial detection was close to the international airport as well. So I think there's some link in terms of the movement and the spread of the beetle throughout the region. Okay, next slide, please. So this is just the summary of what it is at the moment. So we only have about 8 of the Pacific Island countries and territories that are free of CRB. Most of the peaks or the countries and territories are now invaded by CRB, and some of the countries or territories have both the susceptible and the resistant population, while some of them have either of the population. So that is the situation at the moment, and the beetle is continuing to spread. So we may, if they continue to do so, those remaining ones may have the beetles in just a matter of time. Next slide, please. So I think looking at the current situation that we have at the moment, in terms of the spread of the beetle, we really need to have a holistic regional approach to the management efforts that we have across the region. Otherwise, I think at the moment we are working in silos, and if we do that, my view is that the beetle will continue to spread. And we are also concentrating on the efforts within the countries that have the beetle. And for those that do not have the beetle, in terms of their preparations, monitoring, surveillance, we sort of we are not giving much attention to them. So I think we need to the beetle has the chances of continuing to spread into those countries that do not have it. So we really need to have an holistic approach, and then looking at assisting those countries to prepare for any prepare for any potential indication, and have a more an active monitoring and surveillance program in place. And there is also the potential of re-indication into the countries if we concentrate on countries with the beetle, and then in isolation and contain the beetle, the chances of re-indication is there as well. I think that's the case that we've seen for some of the countries which only at one of the populations, and then having the other population coming into the country. And just in terms of the management, I think, we could. This way also touched by some of the presenters earlier in terms of what we can do in terms of the management programs. But I think, in some areas we can look at the eradication. Just thinking in terms of the smaller islands and where the population are more confined. I'm thinking more of RMI situation as well as CNMI. If we want to try eradication, then maybe those are the islands that we can try and look at. But in situations where they're more established, then we look at IPM. And then for those that already have the or do not have the beetle, as I indicated earlier, the monitoring and surveillance program is critical. And also the other thing that's not given much attention is the internal quarantine within the countries as well. We are nations of islands, so once they get into one of the islands, the others remain free of the beetle, and we need to also look at supporting with the internal quarantine to prevent any potential spread within the countries themselves as well. So so what we basically need is that the responses need to be tailored according to the situation. Next slide, please. Okay, so that's just a general situation about the situation of CRB within the Pacific. I'll just now have a look at or run through some of the management programs that we've rolled out within Vanuatu. So the initial indication was on Efate, which is the one with the big red circle, and the beetle that was in 2019, and the beetle was confined, kept on Efate until more recently, where it has been picked up from Santo, just up on the north, the bigger map where the small circle is. So that was just detected more recently in around June this year. And next slide, please. And these are just some pictures of the level of damage that we've picked up in Vanuatu. Next one, please. And the damage were not only on coconuts, but as also indicated by the other presenters for Hawaii as well as Guam. We picked up damage on ornamental palms as well as on food crops like the pineapple here. Next slide, please. So in terms of the management approach, I think we really need to look at it through an integrated approach. I think a single approach won't solve the problem because of the kind of or the situation with the pest. So we really need an integrated approach to effectively control the beetle. So we were looking at awareness, also internal quarantine. There was also a program looking at cares for beetle, sanitation, meteorism, pheromone, and the virus as well as an integrated approach to containing the beetle. Next slide, please. So just in terms of the awareness, I think, yeah, I might add that there was a lot of training that was done to the team as well, because the beetle was new within the country. We had to run a lot of awareness programs, both community as well as general public awareness programs, and also provided a lot of technical training to the team, and there was also some careful engagement of staff that were involved with the program. So we engage staff as well. Next slide, please. Okay, another program that's more an initiative of the local national government within Vanuatu was the internal quarantine program that was put in place. As soon as the CRB was detected, there was a ministerial order that was put up for vessels not to leave in the night from Efate that were going into the other islands. And basically for Vanuatu, the main coconut growing islands were Santo and Molecular, which is also next to it. So they didn't want the beetle to get there. So they had that ministerial order in place. And within that order, they asked for the vessels not to depart after 4 p.m. And also did a full vessel and cargo inspection before dusk. And also at all the lights off when overnighting at the wharf. And for vessels that had lights on, there was also night operation to inspect the vessels to see if there was any beetle that were around the vessels for them to remove and kill. And the next morning before the vessel left, there was also an inspection done to the vessel. Next slide, please. And that just in terms of the results within the last year, the last couple of years, where they inspected more than 50 vessels, and sure enough, they detected around about 20 CRB. There were 7 males and females, so yeah, there were females that were detected. So if they had spread, then yeah, you would have active breeding that would have taken place. And there were numerous potted plants that were checked as well, with some confiscated if they were suspected of containing CRB immature stages. And yeah, the other target islands, particularly the two, remain free of CRB until yeah, the recent detection in Santo. So that's the situation with that operation. And next slide, please. Okay, yeah, Cash for Beetle, that's also the local team's initiative. They just wanted to see if the community would respond and then bring in the beetles, or collect the beetles and bring in to weigh and then get paid for. So through that program, just ran for only about 4 months. And then within that period, yeah, close to almost 20,000 beetle stages were collected within that period. So there was a substantial amount of. Our only concern was people breeding the beetle for that, but through a lot of awareness, the local communities were advised, and they've also seen the impact of the program. So, and there was a lot of beetles for them to collect from the wild to bring in. So they whatever that was brought in was brought in from those that were collected from the wild. Next slide, please. And there was a lot of sanitation program that went to it. So collection of the beetles, and then the dead coconut palms being felled and then and bent. So yeah, and also indicated, I think earlier, sanitation is a critical component. Once palms are killed and the beetles continue to breed in, then the population continue to increase, and then you just have flow-on effect affecting the healthy palms within your area of infestation. Next slide, please. Yeah, the other thing was metricium application and infection. So we build up a unit that's given in there, where we had the box that was built, and then we had organic matter, and just below the organic matter there is a collection bucket that's below the PVC pipe, and there is also up at the top of the PVC pipe cut at opposite sides for the beetle to come in with a pheromone on the top, and then the beetles come in and then eat the side of the PVC pipe and then drop into the container for us to retrieve and kill. So we had one system that was for both the metricium application as well as the pheromone trapping, and yeah. So we had those organic matter that were put within the box were initially also mixed up with metricium before applied, and then there was a monthly application that was done, and also checked for potential infection, and we got good infection within the boxes, and we also picked up infection within the surrounding area. So. Which is indicating that the metricium has been spread within the natural breeding sites. Next slide, please. So these are just some of the summary of the infection data that we picked up mainly from the artificial breeding site. It's quite low, but I think in a way it contributes to the management effort, as we also get in the spread of the metricium within the natural breeding sites. And next slide, please. Okay, yeah, the pheromone trapping, as I indicated, it's also part of the metricium application unit as well. And this is we mainly use the PVC pipe with the collection bucket at the bottom to collect the beetles that get trapped. And then the idea also is that those beetles that miss the PVC pipe and get dropped, they can either breed within the organic matter or be there and then pick up the metricium for infection. Next slide, please. And yeah, these are the numbers that we collected over the last two and a half years. Yeah, almost just over 16,000 beetles that were trapped in the pheromone traps. Next slide, please. And then I just wanted to very briefly highlight on this effort as well. Yeah, our collaborator Ag Research is also was also able to work on the virus as part of the research, and they've done preliminary field releases, and we are at this stage getting some positive results from the establishment within the field. Next slide, please. And yeah, also just wanted to highlight here that as much as possible, we kept away from the insecticide use, mainly due to the safety issues for the contact spraying of contact insecticide due to water table. We decided to sort of limit that, and also at the same time for the TTI, the residue issue, it's really impossible to monitor any potential impact on humans collecting the nuts and having them. So we just kept away from the insecticide application and then just concentrated on the other management efforts. Next slide, please. So that's, yeah, I'm almost there now. That's our last, second last slide. But just wanted to, it's still early days, but we are seeing signs of recovery from on the palms in the areas that we've applied the management programs. And we are hoping that the palms will continue to recover over time, and then we can effectively also suppress the population of CRB through the effort. Next slide, please. And yeah, I think, yeah, it's needless to say that, I mean, it's also important to highlight that those efforts were not with challenges. So I just wanted to highlight some of the key challenges that we had. We lost almost about 2 years of our activity to COVID-19 due to travel restrictions and lockdowns of the program. And Vanuatu also had a number of disasters, natural disasters, particularly the cyclones that went through, that also made the situation worse in terms of the palms that were killed for potential breeding. And also the biosecurity or the internal vessel inspection program that was implemented also at its opposition, especially from the vessel operators, because it affected their program. So there was opposition, but with a lot of awareness, it was eventually understood. And we had also had some vandalism issues with the setups from local communities. Okay, yeah, and on to the last slide, please. I should finish off soon. And I think the last slide, I just, Roland has sort of very well highlighted the importance of coconuts to us in the region, but has also been someone from within the Pacific region as well. I just wanted to re-emphasize and finish off here by saying that coconut is pretty much everything to us. It's our way of life, and it's our tree of life. And as much as possible, we just need to protect the coconut from those anything that would cause the damage to it, including the CRB. And I think that's it. And thank you very much, Andrea.
Thank you so much, Mark. That was a lot of great information. We are ahead of schedule, so we do have some time for questions for Mark. If anyone has, there's already some that are some questions and comments that were going on in the chat while you were speaking. But if anyone in the audience has a question, we do have a few minutes. If you could just use the Zoom button to raise your hand. Let's see. We have one person. Is this Ranu from I think from Marshall Islands? Did you have a question? Your hand is up. Okay. Are we able to, is he able to?
I think in this format, we may not be able to have them talk. So I think it probably, if you have to put questions in the chat or the Q&A box.
Okay. All right. So there, we had in the chat, there was some discussion and questions about the OrNV resistance of the CRB in Hawaii. Let's see. Mark, you had a slide in your presentation that indicated that the Hawaii CRB are resistant to the biocontrol virus. And there was some discussion in the chat about that. Are you able to elaborate on how you know that the CRB in Hawaii is resistant to the virus?
Okay, yeah, if the Hawaiian team has additional comment to make there, that they can do that, but from the information that we have is that the population is related to the Guam one, and I think the Guam one is also from Aubrey's presentation that they've tried the virus and that it is resistant to the virus. So I think that yeah, it is resistant to the virus, and I think there was some from the molecular work that was being done, it is related to the population in Guam. So that's where we are thinking it is resistant to the virus.
Okay.
But if, as I say, if the Hawaiian team has some other information, that can provide that. Mm-hmm.
Yeah, I think there's our next speaker also will kind of address that, and then I know we've got some ongoing projects to examine that more closely with field testing for OrNV and the CRB in Guam, and I think there will be other opportunities during the symposium to talk about that. There was another question, whether there are any parasites. I'm trying to find it again. Where is this? Are there any parasites, for example, parasitic wasps that target CRB? And that question is from William Steiner. Is this anything that you have knowledge about, Mark?
Okay, yeah, with respect to the work on parasitoids as well as the predators, There has been a lot of work that was done with the initial incursion of the beetle within the region that covered both the region as well as the native ranges, and there was extensive search for other biocontrol agents, including the parasitoids and as well as the predators. Tests were done within that time, but most of them were found not to be effective. And I think from that initial study, the two promising candidates that came out were the metricium, the fungus, and the virus, which were also sourced from the native range within in Malaysia. So those were the ones that were actively used, and then so I think in terms of that's the similar situation at the moment with the current invasion that we have.
Thank you. There's one other question that's come in from the chat, Mark. And this question is it possible to use IPM on infested islands to have long-term suppression of CRB? Or should we expect a long-term decline of coconut trees over time, given current suppression tools?
That's a tricky one. Yeah. It's a decision on whether you want to let your coconuts go or you want to protect the coconuts. So if you do really want to protect the coconuts on the islands, then you'll have to go through the IPM to manage the beetle. Otherwise, if you leave it, then you lose all your coconuts, and then you may have to. But then we've noticed that they now have the alternative alternative host as well, in terms of breeding as well as feeding. So it can lead to other food security issues as well. So I would think that you really need to control the beetle and get a population down.
Yeah, I think that is true. And you see in some of the islands, like even in Guam, we've had the beetle. It's for 15 years, going on 20 years. And yes, our coconut trees look bad. In some areas it's worse than others, but overall we still have coconut trees 20 years on, almost 20 years on. And you know, this Guam, you know, is in one of those situations where we don't have other predators that like birds that can help suppress the CRB population, but other islands do have those predators that can feed on the CRB. And I think it's hopeful for other islands, but you will see also in Guam, we have brown tree snake, and since the birds are gone because of the brown tree snake, the tree snakes that are remaining, they're switching to other food sources. So they are smaller, but the snakes are still here, unfortunately, but they're no longer as numerous or grow as big as they did previously. So they're island to island, they'll have to make their choice of and understand what's available on their island. And I'm just looking, I think we have about 2 minutes left in the schedule. I don't know if this is, if we have enough time for one more question. I don't see another specific question.
I think we have to move on.
Okay. Yes. So thank you, Mark, so much for all of the information and for responding to some of those questions before. And great work for everything that you're doing with the Pacific community. And I would like to take this time now to introduce our next speaker, who is Dr. Tek Tay, coming in from Australia. And he's going to be talking with us about a bit more of the CRB genetics and the mitochondrial genome analysis and the haplotypes in CRB. So this is Dr. Tek Tay from CSIRO from Australia. Tek, if you could go ahead and turn on your camera and share your screen. And take it away. Thank you. right, we see your edit screen in PowerPoint. So if you just go to your presentation or presentation mode.
Can you hear me?
Yes, we can hear you.
Okay. Thank you for the invitation to present some of the work that my team and colleagues have been working on. We started this work with a fairly small funding from the Australian government, Department of Foreign Affairs and Trade. So I'd like to take this opportunity to thank the funding support from the Australian government. So today I'm going to talk about looking at the whole problem from someone who had never worked on coconut rhinoceros beetles before, but with fairly extensive experience working on various molecular tools and population genomics on invasive species. Okay, so we all know about the fairly influential paper by New Zealand colleagues, Sean and Trevor and various other people, using the mitochondrial DNA CO1, which is a barcoding gene region, 676 base pair approximately, that identify a unique signature present in Guam. And this signature was subsequently also detected in various islands, and the associated studies where the virus was unable to be established in Guam population, and this was somehow linked to by other studies to the presence of the similar signature across Pacific Islands, and therefore somehow concluded that the other island populations with the Guam signature are also resistant. And over the times since early 2000 to currently, this signature has been detected in many different populations across both invasive and introduction ranges. There have been a few other population genomic studies looking at whole genome signatures, genome-wide signatures, for example, by the Hawaii University group by Jonathan and colleagues, where again, the Guam haplotype or the G haplotype have been identified, again, based on the mitochondrial DNA CO1 gene. And this was detected in, for example, in Hawaii, in Guam, in Palau, and also in Taiwan, suggesting that, again, potentially this signature is being detected across everywhere. However, if we actually look at the same paper, looking at the nuclear DNA signature, the Guam population actually has not been detected anywhere else across all the other sample populations. So this is a bit of a conundrum, because from the same individuals, and depending on the type of markers that one is using, it somehow gave different information and conclusion. So in this study, if we were to go with the nuclear genome, Guam actually hasn't really gone anywhere at all. Likewise, another study from Caven and colleagues from Australia, they look at the population genomic studies using whole genome sequencing approach. Again, there were many different conclusions reached. For example, the presence of the G haplotype on Solomon Islands, which was supposed to be resistant to the virus. However, when they look at the virus genome, they are actually detected in some of the G individuals. And the studies actually had some fundamental issues. For example, there is a assumption that because there had been this unique Guam mitochondrial DNA haplotype being detected and reported, therefore one can apply that previous studies and just assume that that is the same signature being and can be detected across the other Pacific Island nations. In such a studies, it would really be more appropriate to also include the original populations from Guam as well, which wasn't included. So you can see that this study by Atabari et al. Therefore suggested that the Guam signature is also detected in Solomon Islands and New Caledonia and so forth. So based on all this very extensive studies by many different groups, there have been various IPM options being developed. But one thing that is being proposed based on the knowledge that we had before is that one needs to determine the biotype and test for the virus, and that it would be important to match the virus strain to the CRB biotype. So this really requires time and funding, which unfortunately, as we know, the virus, the beetle and the larvae are there 24/7. The beetle will attack the tree 24/7, and funding is difficult to come by. So having this approach, while it may be valid, requires a lot of effort and the time that we don't really have. Furthermore, we know that currently there is no virus that is identified to be able to be effective control for the CRBG. So there is a need to, for example, look for alternative biological control agent such as metarhizium. So what is it that is so difficult to understand or make it clear why we are experiencing such conflicting findings with respect to whether the type of markers we're using, the CO1 versus a nuclear, or whether there should be a specific strain of virus that we should be looking at to target specific population. So I'm going to take it back to the original concept about the mitochondrial DNA genome. It's inherited from mother, has about 37 genes. Some are coding and some are non-coding genes. It is inherited as a single genome, so from mother to her children, and it's always passing on from the female line. So the assumption is that if the CO1 is identical, then the other genome region must also be identical. And if we're looking at here on the top right-hand corner, this is the mitochondrial DNA genome, about 20,000 base pair, 21,000 base pair. And we're only using about 670 base pair. That is, we're only using 3% of the mitochondrial DNA genome to try to track a really, really significant agricultural pest. So here, these are some real data. So this is a Guam individual, which has this specific type of CO1 signature. And if you look at just the 670 base pair region, you would conclude that Philippines, Rota, Hawaii, Taiwan, PNG, Marshall Islands, and Solomon Islands all have a G haplotype. Whereas Fiji and Samoa, they have something different. Likewise for Palau, there's also something different. But if we extend it across to the whole mitochondrial DNA genome, we can see that in fact, this is not the case because we're essentially looking at very short region of the genome that limit our powers of detection and differentiation. So Guam is actually really, really different. There's these two other unique base substitution that differentiate it from everything else. So if that's the case, now not just looking at a 670 base pair region, but looking at the whole mitochondrial DNA genome, Guam actually hasn't gone anywhere. So this is indeed what we did. We look at the whole mitochondrial genome of Guam, as well as other populations from Philippines, Hawaii, Solomon Islands, Marshall Islands, and so forth, and including substantial from native range, such as Indonesia, which is here, and also Malaysia. And we can see that Guam indeed really hasn't gone anywhere. Guam just remained on Guam. Surprisingly, we were able to detect another new populations from samples provided to us just in 2 years ago, where Guam population actually had another offshoot, and this likely represents a very recent introduction. So this shows that populations can be quite mixed and have very different origins. They're not just a single population. And that in reality, if Guam is a concern because it had not been able to have the virus established, then Guam hasn't spread anywhere, then how do we actually reconcile the widespread reported severe damage and control failure when the countries are mistakenly informed that they have the G biotype? So that's something that we actually need to address. And I think that that's probably something to do with the fact that it's our ability to properly identify the populations, and we're giving the wrong advice. So you've seen this from Mark's presentation earlier, but if we were to reassess using the whole mitochondrial DNA genome now, we can see that the pathway is actually really, really different. So for example, Sri Lanka has been suggested as the origin for Samoa and Fiji, and in fact, that's the case. We could link the population from Samoa and Fiji directly to Sri Lanka. We can also identify Malaysia to have been responsible for example, Solomon Islands, and their link between Solomon Islands and Papua New Guinea. And in fact, Solomon Islands population was also linked with Marshall Islands, which was the most recent introduction. Palau had both Philippines and Indonesia, and Rota and Hawaii, for example, as well as Taiwan, had Philippines. So a summary from that, from the presentation that I just spoke about, using PCA, we can see that the green boxes or green highlighted populations are native, and the salmon color countries are introduced ranges. And we can see that there are multiple populations, such as Malaysia, that's being detected elsewhere in different places. Philippines and Taiwan appear to be responsible or very tightly linked with a lot of invasive ranges. Likewise, Malaysia, and even within China, it's their internal movement within native ranges as well. And we can see that these are actually the biosecurity hotspots. So Philippines, Indonesia, and Malaysia appear to be the hotspots for a lot of the Pacific Island nations, CRB populations. And this is important because we have another really important pest group, the wrinkled forest palm weevils. They are in the same native range as the coconut rhinoceros beetles, and these are major pests for example, coconut as well as palm oil. And if coconut rhinoceros beetle can get to the Pacific using this weak biosecurity measures, options, so can these other invasive weevils as well, right? That's what I think. So if we are still really quite keen to track the Guam population, which is important because we know that the Guam and the Guam population has been reported as resistant to the virus. And as Mark also pointed out that, you know, other populations are known to be resistant populations such as the Philippines and Indonesia. And we have identified that these are the biosecurity hotspots. Then we really need a very effective marker to track the Guam population. And we have actually working with New Zealand and Indonesia, as well as Philippines and Palau and Guam. We have developed an alternate set of markers, which combines two genes to differentiate Guam away from the other populations. Whereas if you use a CO1, you are unable to properly differentiate the Guam population from the rest. So this is currently it has been revised and resubmitted, so hopefully it'll be accepted soon. So going a bit forward in terms of what we understood with the genome signature now, and addressing the question about, do we need to match the CRB biotype with a single OrNV strain? We know that in the past studies, for example, by Zelensky and colleagues, they have undertaken this type of experiment in the Maldives. They've carried out experiments involving either mixed or single isolates. And they were able to demonstrate that, at least the report, that the most effective isolate was an isolate called X2B from the Philippines. However, even though this is a claim, but if you read the paper, there's actually no detail about how this X2B is reconfirmed as the isolate being the most effective. Also, in some cases, X2B were introduced, however, unknown isolates will recover, as well as there is a huge high mutation rates based on what we currently know from genome sequencing of the virus. And so this can rapidly lead to mutation. And so there are a lot of issues that may be associated with just introducing a single strain. So again, I'm just going to reiterate that for the case in the Solomon Islands, there's been talk about the need to test the range of different virus strains to hopefully identify one that is effective against a G. So just bear in mind that as the whole genome, or at least the whole mitochondrial DNA genome, evidence is suggesting that Solomon Islands at least doesn't have the CRBG. And do we really need to go ahead and do individual strain testing? I think that that is up for debates, but based on previous studies, it would seem that if you introduce multiple isolates, perhaps most effective isolates be X2B or something else may emerge anyway. And that's necessary because if we look at the published virus genome studies, there's been two very good study, very timely studies that have been published, one by New Zealand and one by Australia. And if we look at one of the X2B isolate that's been published, the genome signature is really, really difficult to interpret, and we can't really confirm with all these different single nucleotide polymorphism dotted across the genome to really use that specific polymorphism to say we have a specific isolates. Furthermore, there are many genes, over 130 genes, potentially in the virus genome, and not all genes' functionalities are known. We also do not know that in an infected CRB, at least in the native range, whether this is actually by a single isolate or is it by many different isolates. And what about in CRB populations where there are multiple origins? So there are multiple biotypes, if you like, or biotypes doesn't really mean anything, just where there is higher genetic diversity in the introduced population. So which isolate do you actually need to use to cover all this different genetic variability within the host population? So these are questions that I think we may be facing if we're trying to choose to identify a specific isolates of the virus to use. And finally, I just want to quickly show in the last two slides that we actually look at the genomic basis of the virus infection. And we have Indonesian populations all across here where we have confirmation by visual dissection. showing that these are infected individuals versus healthy individuals. And you can see that the whole genome sequence reads are high, over 1 million reads, most of them. And in this case, where this is representative of a natural population, we don't just have one isolate. We have many different isolates under natural conditions, and we should be really pushing to repeat this type of approach or the condition for the introduced populations. So in Indonesia, if you look at the Indonesian populations, overall, they are the same isolate, if you like, but they're just different genetic variation across. So not just one variation. Likewise, for the others meaning representing Malaysia, as well as the other Pacific Islands, there are many different isolates, if you like, very high genetic variation. So we shouldn't be just looking for one specific isolate to try to address the CRBG. And likewise, we didn't actually know a lot about the genome basis. So in some cases where the dissection indicated that the CRB actually has really healthy guts, we're able to detect a lot of virus sequences in there as well. So these are issues that we are still currently working to address it. So my last slide look at the implications on the IPM strategy that we're trying to develop for the CRB, based on what we currently know, based on whole genome approach. That is, if we're trying to match a specific biotype with a specific strain, why are we doing this now when in the past, as well as under natural conditions, this is not the case? So we need to really think carefully to see how we can repeat earlier successes that have been trial and shown in, for example, in Fiji and the Maldives. So again, the original Guam CRB is not present across other Pacific Islands, at least not during the time that we did the sampling 2 to 3 years ago. So how is it that when CRBG is misidentified, it is then supposedly causing significant coconut palm damage as well? So that's something that we really need to think carefully and try to work out why is it that we come to the conclusion that once a country has been told that they have the CRBG, they are also reporting that they have this huge amount of damage. Is it because of specific IPM solution that is not properly developed or suboptimally developed? So we really need to reevaluate current IPM approach, as well as increase our understanding of the genome of the virus. And the next talk by Ben Hoffmann will talk about the importance of improving phytosanitary practices. But I was just want to highlight that this is really important, and we need to work that together with a genome approach to detect new introductions. And maybe we can use that as a solution, because if a virus is naturally introduced the new introduction populations, then the virus is there. For example, this may be the case that might go forward for Hawaii, for example, if you have something that's difficult. So if you can really detect the novel introductions, but potentially you can actually get the virus naturally coming in, and you can identify the populations properly and utilize that that resource that is coming up from a bad situation of introductions. Thank you. So this work was again funded by the Australian Department of Foreign Affairs and Trade, and we are not able to do all this work without all the international support and colleagues. Thank you.
Great, thank you so much, Tech. Oh, sorry. Great talk. This was a lot of information on the genetics of the CRB and also the OrNV that's been detected in the CRB genomes or specimens that have been sequenced. There were a number of questions and comments in the chat while you were speaking, and we do have a few minutes if we could go ahead and address some of those. And one of the first is from James Grisella. This is Jim. He says, it seems like we have to evaluate the use of the terms CRBG, CRBS, and CRBPNG that supposedly designate susceptible versus non-susceptible CRB populations. So there's no question here, but how would you, based on all of the genetics work that you've been doing in the last couple of years, how would you recommend or suggest that the different biotypes or different genotypes start being referred to?
Yes, thank you. Look, well picked up by the person. Sorry, I missed the name. Jim.
Jim.
Yep. Look, I truly believe that you're spot on there. We shouldn't be using the old names. We should at least move away from referring a population and its assume status, resistance status to a specific haplotype. We shouldn't be using the term biotype either. So in terms of how do we address it, I think similar to the whitefly scenario, you might be able to address it by the location where it came from, but certainly you should not attach resistance status to any of the population unless they're properly tested. I have no solution for naming it. I haven't thought about how to name it. What I'm thinking about is how to actually resolve the many different conflicting results coming up from the use of partial genome or partial gene as markers, and the different proposal of IPM solutions. I'm just trying to think in a more holistic approach. So yeah, I don't have an answer for that now.
Okay. Yeah. So just some comments in the chat about what excellent insights and great presentation, and the way, very good job on explaining all this complex information. There was another question from Jane Beachy. And this may, you may be able to address this, or we may actually ask this again during this afternoon's open Q&A panel. But from Jane Beachy, she asks, besides the difference in susceptibility to nudie are there other differences between CRB types, such as habitat preference, tolerance to cold, heat, preferences for relative humidity, or reproductive success? So I don't know if you'd be able to address that, or if we move that to this afternoon's panel.
Perhaps to the afternoon panel. I mean, like, believe it or not, I actually, because I live in Canberra, Australia, which is inland and half in the altitude, and it's cold. I don't actually come into contact with Coconut Rhinoceros beetles altogether. But yeah, look, I think it's very interesting, and it's really, really important question, because under the current climate change scenario, all this, different habitats, microclimatic, I guess, environment, and all the other related ecological questions to the actual test will be really, really important. But at the moment, I probably can't answer that.
Okay, well, we'll add that on for possible inclusion in this afternoon's panel. So we're at 10:18 this morning. Sorry. So we are scheduled for a break right now. So we have a 5-minute bio break. But in the meantime, during the break, we have a short video that Aubrey Moore, who presented yesterday, he's offered to show during the break time. So if we can get that queued up, is Aubrey on the line to share his screen for the video?
The answer is yes and yes.
Okay, great. So we'll be meeting back here at 2:25 to continue on with the next session. But in the meantime, here's this video from Aubrey Moore.
Yeah, so yesterday I mentioned that there might be a problem with interpreting trap catch data, and I'd like to explain what I was talking about. So let me share my screen. Okay, can people see my screen?
Yes.
We can see it.
Fantastic. All right. This is a very short presentation. Let me just get to the slide that I showed yesterday. Okay. So this is the slide that I showed yesterday, and it indicates that population density, trap catch is not a linear function of population density. And why might that be? Well, this comes from an understanding of how pheromone traps work. And this is basically a theory of mine. It's not a fact at this point, but it's a very simple concept. All right, sorry about this. Okay, so the usual assumption is that trap catch is a linear function of population density. And that's quite reasonable. You would think it would be. But let's have a look at how pheromone traps actually work. So here we have a representation of a pheromone trap. The red dot is the lure. And then there's a plume of molecules coming off that lure. They're blown by the wind. In this case, the wind is coming from the north, blowing towards the bottom of the screen. And the way insects navigate these plumes is they fly across the wind from left to right or right to left. And if they find a pheromone plume, they will immediately turn and fly upwind. So if an insect is flying across the screen at the bottom half of this representation, if they hit one of those white dots, they'll turn immediately upwind and fly up towards the lure and get caught in the trap. All right. So what happens with CRB is the lure is actually a synthesized form of the pheromone that's produced by the males. So if you have a population that has males in it that are producing the same pheromone, there's going to be competition. So let's put a couple of green dots here. The green dots represent male CRB adults, and they're putting out their own pheromone. So if you have a male or female insect flying through the bottom half of the screen here, they will first encounter the plumes coming from the natural sources of pheromone, the males. So they'll turn, fly upwind, and go and find the males. So there's competition there. Even at low population densities, there's competition for that pheromone. What happens when we have a high population is much worse. You not only have competition, but you have total confusion. There's so much pheromone out there that the insects cannot navigate to the source, either a natural source or an artificial source, a pheromone trap. So at very high populations, the traps may not catch anything at all because there's all of this other pheromone out there. We actually use this phenomenon to control certain insects. There are lots of moths that can be controlled by just disseminating pheromone all over the place. It's called mating disruption, and it's very effective with some species. It could be used for rhino beetle, but it's never been tried. Okay, so given that demonstration there, you can come up with a curve that looks more like this. At low populations, the traps work perfectly fine, and there's an almost linear relationship. Then you get to a high population, and at some point, the line starts going down, which is not intuitive, is it? Your population density is going up, but your trap catch is going down. And I think it can actually go down to 0. In fact, I think I've seen an instance where it did go down to 0. Anyway, is there any evidence for this in other species? And yes, there is. I came up with a paper that where they had a measure of actual population density of forest tent caterpillar. And they measured their trap catches, and the trap catch actually gave a curve similar to what I showed you in that theoretical model that I just showed you. So that's all I wanted to do. It's not a fact at this point that trap catches at high population levels are not correlated, but I think we got to look into it. If we're very lucky, all the population densities that we're looking at with CRB might be in the first left part of this graph, and there is a correlation there. At least if the population density goes up, your trap catch goes up instead of down. So that's all I wanted to say. I just wanted to clear that up. Thank you very much.
Thank you, Aubrey. I'm wondering, the video that you were showing with the interference from the other natural sources of pheromone, is that also a partial explanation why we see a slightly higher rate of females in the trap catches than males? It's not, it's like...
Yeah, so the pheromone we're using is not a sex pheromone. It's an aggregation pheromone, and it attracts, as far as we know, it attracts both males and females equally. I don't know if the difference in trap catch between males and females is quite small. It's less than 10%.
Right.
Like the first cut is that, you know, the population is 50-50 males and females. So I really don't know if the actual population has more females than males or not, or if this is an artifact of the trapping. I don't think anybody knows that. We don't know it with the Guam population, especially.
Great. Thank you so much, Aubrey, for that short info about the population density versus trap catch. I think there's some questions that maybe we can shift to the open panel Q&A this afternoon. We hope that you'll be able to join us then.
Yeah, I'll be around.
Okay, great. So if you wouldn't mind, stop sharing your screen.
Yeah.
We are ready for our next talk. So we're starting the next session. Franny, are you carrying on with this, or shall I go ahead with the introduction?
Either way, I'm good with it. I think we're going to go right into it because we're just about at exactly the right time. So happy to carry this on. I was really caught up in Aubrey's presentation as well, trying to figure out we have a lot of questions being in an early detection phase. We have a lot of questions about the effectiveness of traps. So that was very interesting. Thank you so much, Aubrey. Yeah, okay. So for those that missed Aubrey's presentation, his little mini presentation there, I did put the link in the chat, and he's got some AI voiceover to entertain you. So if you did need to use that break to take a break, that's all good. You can follow that. And of course, you can put any questions that you have there in the chat. Thanks again for everybody who presented. As we move on, we are going to be going from the very minuscule, looking deep into DNA. And we're going to go way broad for the next section of today and be looking at biosecurity and interceptions and movement of CRB and how we can interrupt that movement and detect. We've had a lot of information today about how the beetle is moving or maybe moving across the Pacific. And we know there's still a lot of places that are not infested, like I like to consider the island that I live on not infested. So how do we prevent those areas from becoming areas that have to deal with CRB? So really grateful to everybody who's here today for that. And we're going to start with, I think, a colleague of TechTay's there, Dr. Ben Hoffmann, who is Principal Research Scientist at CSIRO Health and Biosecurity from the Tropical Ecosystems Research Center in Australia. Ben, if you'd like to start sharing whenever you're ready, and we'll be very interested to hear what you have to say.
It's Franny. Hi, everyone. Can you just confirm that that's showing properly?
Yep, looks great.
Excellent. So, hi, everyone. For those who don't know me, I'm Ben Hoffmann. I'm actually an invasive ant researcher, but I dabble in all sorts of biosecurity matters. And over the last few years, CRB has got my attention. And what I'm going to talk about today is a paper that's literally just come out this week, myself and some colleagues, about biosecurity interceptions of Coconut Rhinoceros Beetle. And this came about, I think, because one day I was just pondering what do the data look like for this species of the biosecurity interceptions. And I think what I had in mind was Australia is still free of this species. And so we really do want to know this information if we're going to keep that status. And when I went to search for the information, I couldn't find it anywhere. And it looked like nobody had published on it before. And so that started a quest to try to find that information. And in doing so, I ended up with a couple of collaborators, and we've now come out with this paper. Oop, now I've got to work out how to change the slide. There we go. So the primary places we went looking for were the biosecurity databases of locations that do regular inspections. So it's very well quantified. And we managed to get this from 4 databases, one being Australia with 2 decades of data, New Zealand, also 2 decades of data, the European Union and Switzerland, which is about 15 years, and from US Defence, the contractors doing cleanliness inspections of goods being moved out of and into Guam and Rota. Of course, we wanted as many as we could get, but there were a few that we couldn't access. And an example of that is the biosecurity database of Fiji. They do have a really good one, but we couldn't access those data for whatever reason. And we also know that there are inspections happening in places, but there's no particular database for it. And an example of that is Guam civilian ports. So to extend our data, we also did general web searches. And in particular, we found one publication, Kumar and Bigsby. It was a pest risk analysis of US ports, intercept data of about 15 years up to 2014. And we also found publications that had 4 more interception records. And at the end of the day, we had to draw the line somewhere, and we pretty much exhausted our searches at this point, but we decided that it was sufficient to give good insight into what's happening with CRB. I want to be very clear exactly what we didn't do. We didn't focus a gathering of data on the ports of exit. We weren't really concerned about where the beetles were coming from, and partly because we didn't want to make this into a political document because there are issues for trade. And so along those lines, we also didn't do any risk assessments. So that's a completely different science that somebody else can do. So what have we found? We got 52 confirmed intercepts. For the 2 decades for New Zealand, they got it 13 times. In Australia for 2 decades, they found it 14 times. In Guam since 2017, it's been found twice. And from the USA up to 2014, it was found 8 times at ports. We found no credible interceptions from the European Union. So there were beetles reported, but it was typically just something like a rictus spit. And so we discounted those. And there were 4 intercepts reported from 2 other jurisdictions. Now, the only picture you're going to see in this presentation, this is what that all looks like cumulatively. So the bottom sets of patterns there are the major countries, their individual data, and then the top one is all of that combined. And what you can see is there's a pretty constant rate of interceptions, regardless of whether you're looking at a jurisdiction or at the global level. There might be something interesting happening there at about 2009. There does seem to be a little bit of an acceleration, but we couldn't ascertain anything from that. And maybe that's a good point for somebody else to look into in the future. But otherwise, it's pretty constant. We then looked for any patterns through those data that could help for biosecurity. And unfortunately, there really wasn't much at all. All but one of the detections were adults, and of those that were sexed, they were pretty even sex ratios. For in terms of month of detection, there was no seasonality found in any of the detections, predominantly from the Australian data, which included month of detection. Most of the beetles found were alive, and so these beetles were capable potentially of inducing new incursions. And maybe there's a story from New Zealand with their interceptions from the Philippines. What we found is that there were regular occurrences from the Philippines. This is the one and only case where I'll mention the source. And then it suddenly stopped. And there hasn't been an interception for something like well over a decade from the Philippines in New Zealand. And we actually contacted the New Zealand authorities to ask if there was a protocol put in place to make this happen. And unfortunately, nobody knew the answer, and I suspect that there probably was, but the people have moved on. But in any case, there is another very interesting thing for somebody to look at with these data to work out why New Zealand is no longer getting these beetles when they were in the past. Probably the more concerning thing is that the detections were associated with a very broad array of commodities or situations. So here is the list of things that they were found in. I'm not going to go through them, but needless to say, they were found in all sorts of vessels, including an aircraft cabins and the hold and their wheel wells, all sorts of producers, and even concerningly, a fair few were in the aircraft passenger luggage. This is just in their bag with their clothes. And with a bit of investigation with this, it was clear that people weren't deliberately bringing the beetles into places where they were going to. The beetles were literally somehow getting into their luggage. So what does all of this mean? Well, unfortunately, CRB appears to be an extremely generalist passenger, and it's capable of being accidentally transported by a wide variety of means, literally at any time. And what it means is that there's pretty much no possibility of us focusing on any particular commodity, packaging, vessel, season, etc. To detect this species. And it's simply going to come down to really good biosecurity vigilance on all goods and vessels arriving at and leaving destinations. And that's it. Thank you very much.
Wow, that was fantastic. That was fantastic. Thank you so much, Ben. I feel like I'm learning so much from these talks. And one of the things that has been a really big deal for us is trying to figure out which commodity we really need to focus on. And obviously, everyone, green waste and mulch, but, from what you're saying, it's really, there's a lot of potential weak points that we need to be highly vigilant about. And that's very helpful information. So Laura Brewington says in the comments, CRB in the clothes, no thank you. And I think we can all get behind that. I'm trying to see if there's any questions that we want folks want you to answer right now. You're getting
in one of the slides that Mark showed of the beetles literally flying onto the ships at night time, and the number of interceptions there. That's scary, knowing that the beetles are just flying onto the decks of ships and the ships are moving on and going elsewhere. The amount of vigilance that is going to be required to stop an incursion occurring like that is just incredible.
Yeah, I think that really echoes back to Mark's presentation this morning, talking about, how much security they're trying to take with those ships moving at night, and they have to turn all the lights off. You can't, have any lights on if you're on the ship. And I mean, that is a really difficult level of security to maintain. And kudos to anyone who's able to do it. That's a lot of work. Christy's saying, great talk. And did you report on living/not living? And I think I did see that on one of your slides, how many beetles you were finding dead versus alive.
Unfortunately, so a high risk of starting a new incursion. Sorry,
that you faded out a little bit at the beginning there. What did you say?
Correct. So they were mostly alive.
Mostly alive. Okay. That's upsetting, honestly. Okay, let's check our time. Right, I think we want to move on. Actually, we do have some time if anybody else has a question.
I can see Aubrey has his hand up.
Yes, I do. And I do have a question and a comment. I have some records of interceptions which aren't on your database. My question to you is, are you going to curate a database ongoing? Or is there a way of doing that?
No, but in the paper, we've provided a summary table of everything that we found. So that could certainly be a basis for that.
Fantastic. Yeah, I'll get in touch with you about that. Just of interest, there's two interceptions on Saipan, which is an island just north of Guam. And one of them was a beetle that was caught there a year before it arrived on Guam. So that was an early warning sign. And the second interception was actually when several pallets of soil, packaged soil, were shipped up to Saipan, and they were full of grubs and adults. And then there's the Mexico interception as well, which you probably know about. Yeah, thank you very much. That was really interesting stuff.
Yeah, it was. And I see another comment. Someone's commenting, I think this is from Dan, your presentation yesterday, that some lights actually repel CRB. So, my gosh, another layer of complexity. What type of light are we talking about? And some attract, some repel. Very problematic, very problematic. Thank you so much. If anybody has any more questions, please go ahead and post them, and be sure to stay tuned. I hope you'll stay with us then for this afternoon for our panel discussion. Okay, next up, we're going to have Dr. Julie Lockwood, we're jumping to another side of the world from Australia all the way to New Jersey. So Julie is coming to us from Rutgers University, where she's a professor in the Ecology, Evolution, and Natural Resources Department. And she's also the interim director of the Rutgers Climate and Energy Institute. So welcome, Julie. Thank you so much for being here and sharing with us about environmental DNA. I know that's something that everyone's very interested in. I see you have your screen shared, so you can take it away. We're not hearing you though, so.
Yeah, I know. It's like, how many years do you do Zoom and you still can't unmute yourself? But you can see everything okay?
We can see it. It's beautiful. Thank you.
Okay, great. Thank you. So yeah, I'm in New Jersey, so it's quite late here. It's a great group of people. Clearly needs to be virtual since we're doing it from around the world. So I'll talk to you about our work on developing an environmental DNA tool for CRB. And this is a group project with Kathleen Kyle, Anthony Vastano, and Ann Nielsen, all here at Rutgers. So if you're familiar with eDNA at all, you probably were introduced to it by thinking about aquatic eDNA. So, sorry. There we go. Aquatic eDNA. And the idea with all eDNA is that anything that's alive is shedding some form of DNA into their surrounding environment. And so if you can just then grab something that used to be called bulk DNA, but I think it's sort of expanded beyond that. But the idea for aquatic DNA is you just need a little bit of water, and there's a lot of DNA floating around in that water from all the things that were living around in that water. So then you isolate that DNA and you move it through sort of pretty standard genetics tools to figure out what species belong to that DNA. So you can sort of grab one little bit of water and tell all the fish in this particular case. And so that's kind of how it got started. It's been moving pretty regularly out of aquatic world, and our lab has been pushing it into more terrestrial systems. And in particular with forest pests or agricultural pests, all insects. And so we started out developing this for brown marmorated stink bugs. And we've been deploying it, which I'll show you a little bit later, on spotted lanternfly, and lately on emerald ash borer, Asian longhorn beetle. And so we thought maybe it would work with coconut rhinoceros beetle. So in this case, of course, you're not going to grab water. The DNA that's being left, at least for insect pests, is that they're leaving it on their host plants or host trees in this case. And the question is, how much DNA are they depositing in those areas? Where on the tree are they depositing it? That becomes really important. And then how do we access it, since we can't just kind of, it's not water, so you can't just sort of go and grab that. So I'll show you how you do that, but this is the idea, is that in this case, coconut rhinoceros beetles are going about their life and they're leaving their DNA around. And so if we can capture that DNA and move it through the sort of genetic pipeline, then we can confirm that they were there without ever having to catch them. So one question I regularly get is, how do you know the DNA is from the species that you say it is? And the answer to that is that that's a good question, and that's a constant sort of process within the environmental DNA community, is to figure out what part of the genome is best for attaching species to the DNA you get from any one sample. So in our case, we're not doing multi-species eDNA or metabarcoding eDNA metabarcoding. We're doing a more conservative approach, which is using qPCR to identify one species, which in this case is the coconut rhinoceros beetle. So what we're doing with environmental DNA is trying to find trace amounts of DNA, which is usually small fragments of DNA, where we can say, oh, that series of ACTs and Gs over that fragment is going to belong to CRB and nothing else that co-occurs. So the first thing that we did was to develop that assay. So we used some existing genetic information, and then we also sequenced a bunch of coconut rhinoceros beetles as well that were provided to us from our partners, including Keith and Mike and some other folks. And what we developed was a 64 base pair amplicon within the CO1 mitochondrial region. If you're interested in using it, that's the sort of, you can find it using some of that information, but you can also contact us and we'll give you all the recipe and information that you might want to use. We tested it both in silico and in situ to make sure that that fragment of DNA, that 64 base pair fragment, will not amplify in a qPCR machine for any other species. I can't say that for the whole world, but I can say it for Oahu pretty confidently. So we tested it against co-occurring or other beetles, including flower beetles, things like that are common. We just also did some collections while we were in the field to make sure that there's nothing that was co-occurring, and leaving their DNA around would just amplify in our qPCR and indicate that CRB were there when they really weren't. So we had no indication that was true. We think this assay is only going to report that it, that in a qPCR machine if it's really CRB. One of the other standard protocols you use for eDNA is to calculate what's called limit of detection. And it's just a series of in-lab experiments where you figure out how little DNA do you need to have in a sample before you cannot detect it anymore. So these are quite small. So we calculated out that you can detect within a qPCR machine using 3 technical replicates in a qPCR. For those of you who are way into this, about 550 femtograms per reaction, and that the lowest concentration you can detect from using serial dilutions is around 663 femtograms. And just as a reminder, a femtogram is 10 to the negative 15 grams. So what that means is that we can detect very, very trace amounts of CRB DNA in any given sample that we take. So it's very sensitive and so far at least species specific. Here's our field methods. They're very, I don't know, when people think about eDNA, they think that you're doing something really kind of special outside, but really we're just taking paint rollers and looking like we're painting the side of a tree. We look a little nuts when we're out there doing it. It prompts a lot of questions from the public, which has been entertaining. But the idea here, our original idea, which we've used for other species as well, is that in forensic eDNA or forensic DNA work, they just take a swab and they'll sort of swab a table or something to get the murderer's DNA, right? And so what we needed was a really big swab to run across the surface where we think the DNA is. And so what we decided to do was just take paint rollers and make those into giant forensic swabs. And so what's happening is we, of course, sterilize these before they come out into the field to make sure there's no DNA on them. And then you just stick them on a roller and you moisten them a little bit and you roll them on the side of the tree where you think the pest insect is likely to have deposited some DNA. You can use one roller and roll 2, 3, 4 trees. Usually we've done some experiments saying that beyond that, you probably are doing more, you may be losing some DNA. So 2, 3, 4 trees with one roller. Once you've kind of done that, then you take the roller off, put it in a bag, a sterile bag with some DI water, and you just sort of clean it off kind of the same way you would a paint roller at the end of the day, painting your house. So what you're doing in that instance is you're taking any DNA that's on that roller and kind of moving it into solution into that water. After that, we can just borrow all the existing standard protocols for aquatic DNA. So for example, that last picture is you just take a, oh, it's late and I can't remember the name of the pump, but it's just a pump that pumps out the water, moves it across a filter that collects the DNA and a lot of other stuff that happened to be on that roller. And then once that happens, then you take that filter, in this case, these are Smith root filters that are self-desiccating, so you can just put them in a little bag and we can send them to New Jersey if we have to. But you can also take that filter out and preserve it and put it in a small tube, say in ethanol. And then that's it. So the roller doesn't matter anymore, the water doesn't matter anymore. Everything that you need to know is on that filter from then forward. You can see in this left one is that we were on a golf course surveying for, this was, I think, Ted McElwain golf course. And we just had our whole setup on the back of a golf cart. So it's pretty portable. Once you get the filters, you take them back into the lab and you run through the qPCR machine. This is another example. This is stuff we've been doing for the New York State Department of Transportation, sort of following the spread of spotted lanternfly across major roadways. And this is another example of, I guess, basically the low-tech nature of collecting these things from the field. So this is just in the back of a truck. And that's pretty much all you need. So it's not too burdensome. We'd like to make it less burdensome. The other thing we tried was using eDNA to detect the grubs and mulch. So you could just treat this like a soil sample, but there's so much mulch you'd have to go through to figure out whether or not there are grubs there. So we tried something else, and that's just taking some mulch samples, throwing them into one of those bags, kind of making mulch tea, if you will, and then sucking that water out and moving it across the filter. You can see, of course, this is way, way, way dirtier and muddier in terms of what goes on that filter. But it was something we would try because we could get through a whole, a lot more surface area of mulch by doing that rather than just taking the soil and taking it back. That's a very burdensome way of doing that. So here's where we did sort of this in 2 runs. And the first one was just to go out to places where Keith and his crew told us there are most definitely coconut rhinoceros beetles in these 2 places, because we needed to confirm that if they were there, we could detect them. And so in 2022, we did this in 2 locations. My stars are a little off, so you can kind of see where they are. But it's in the Cunha area and then the golf course near Honolulu. So I'll just cut to the chase there, and we could detect them. It was not a big deal. So we in both the mulch and on the trees, we could detect CRB from our rollers and our little mulch process. So that confirmed that the basic idea was sound. Then in 2023, we did what I call the challenge surveys, and that's we went out and did surveys in areas where there were much less CRB, at least in 2023. So some of those places where we have some trap detections, and some places that really weren't, or just a few. So we're just trying to see how well it performed in areas where there weren't a lot of coconut rhinoceros beetles. The places where there were not that many of them, they performed well. We're kind of still working on that data. It's tough data because you have a whole bunch of zeros. So how do you analyze a bunch of zeros? So we're kind of moving on from that and seeing if we can't build that up a little bit better with a follow-up grant. But the one area where we could do a straight up comparison between our eDNA detections and traps was in Honolulu, kind of in the northern part of, in the top sort of star in the graph there. So we just recently calculated this using probability detection formulas from occupancy analyses. So this is a pretty standard way of dealing with probability detection, including with eDNA. We have the site, we needed a site where we had some detections and non-detections all within the same period. And so that kept us in that sort of medium density area. You can't have all ones because then you can't calculate anything, and you can't have all zeros because then you can't calculate anything. So I'll just say that this is kind of a first glimpse at what this looks like, but it's a limited set of data. We really kind of need to build that out. What we did was for the site, when the 1 or 2 days that we were out doing our roller samples and for eDNA, we picked, we got trap data from Keith. In the previous 2 weeks and the 2 weeks after we were there. So kind of bracketed in a month where we were kind of in the middle, and took that trap data and calculated probability of detection and compared to that what we got for eDNA. And the results are that they're fairly equivalent. They're giving you the same probability of detection. So these are the panel traps, which I'm sure Keith can tell you a lot more about. I believe they're baited with pheromone. They're all within a kilometer radius of where we took the eDNA sample, so they're not crazy distant, but they're a little bit distant. And we're basically getting the same kind of return for what we're in terms of probability of detection of CRB if they're there. The big thing, of course, is that we were there for 2 days and then the traps were deployed. We just took the 2 weeks, that's a pretty big difference in effort. The mulch, I'm not going to spend a whole lot of time on these results, except to say that when we looked in 2022, and we were in the high density areas, and we really were trying to figure out whether or not we could get anything with the mulch. It's like, yes, you can detect CRB using our methods with our little mulch tea without much problem when they're pretty common. But when we challenged it and sort of went into places where they're not that common, there's a huge variance in whether or not we would get detections. That's almost certainly from some low sample sizes. But the other thing is that you just can't cover as much surface area in the mulch with the way we were doing it as you could with the rollers. You can get a lot more sites that might have the DNA because we don't know exactly where that DNA is. We're making a guess based on the natural history of CRB. And an educated guess, but there's just the DNA that the grubs are leaving, or it just stays local. So if you don't happen to dig that part of the pile, you're not going to get the DNA. So it's a promising result, but I don't know if it's really operational. So I would say, that's a kind of interesting question about what to do next. My guess is it wouldn't be what we did. Our solution was not the optimal solution, but there could be some other things that we could do, including potentially using air samplers. So how close to operational are roller CRB eDNA surveys? I would say for anything that's truck accessible, where you could get a truck or a golf cart out, they're ready to go. I mean, we've been using the same approach in New York State, following the dispersal pathway of spotted lanternfly, which is very much along interstate highways. And this is just a map of all the areas we've covered in about 4 weeks. We can cover a huge area in just a couple of weeks from the back of the truck. So the efficiency with which you can get to a lot of different areas and knowing that you're getting the same probability of detection, at least for traps, means that I think you could deploy it pretty much immediately. I think it's also ready to deploy in sort of native forests where maybe you don't want to hang a lure. But there are some logistics things like, do you have to pack in rollers and get them out? And so there's some DNA preservation issues there that we'd have to do, but I don't think any of those are insurmountable. The one question that we can't quite get to, but hopefully we're going to get some more money here soon, is to see how well eDNA performs in detecting CRB when they're very rare at a site. So just getting there, early detection, early detection. And for that one, there's a lot of stuff we kind of need to think about. We kind of need to interact with folks who know a lot more about what the beetles do. Where are they leaving their DNA? And maybe more exactly, because now we're talking about only a few individuals leaving DNA. So we have to be very precise in terms of where we go to try to quote capture it. What is the behavior of a rhinoceros beetle that suddenly finds itself on a new island? Are they going to immediately go to a top of a coconut tree, or are they going to do something else? Because if they go immediately to the top, we can target that area, maybe with drones. We've been doing some work with using drones in canopies, tree canopies. And so that, given all the work I've seen in the past couple of days on drones, maybe there's something we can do there. Anything we can do to kind of increase the capture probability or the detection probability this kind of gives us a new tool in our toolbox to make that happen. And then finally, I really, really, really want to combine using pheromones with eDNA, where the idea here is that, and forgive me for my bad PowerPoint things, but when a pheromone trap is luring in a coconut rhinoceros beetle, for you to know that it got there, it has to encounter the trap and then get caught in the trap. And from what I know of other insects, that capture rate is pretty small. So if they're hanging around the trees around that lure and leaving their DNA, then if we just roll those trees around it, then we can effectively increase the capture rate of the trap by allowing ourselves to capture their DNA instead of capturing them. So I would love to be able to try that. Every other insect that we've been dealing with, pest insect, does not have a great lure. So it really wasn't worth trying. But what I've heard so far with CRB, I think it might really work well. So you're just combining pheromone. You might just want to keep the trap out there for regulatory reasons because you need a body if you're going to do anything. But just seeing if you can attract them to the area and then use eDNA to find their, use the rollers to find their DNA. So this, I want to acknowledge that this was funded from the U.S. Department of Defense and their CERDIT program, and this is our group. And I also want to have a huge thank you to Mike Melzer and Keith Weiser and his crew, because they hosted us and allowed us to send stuff from New Jersey. Made our life a lot easier for doing that. So that's it. Thank you.
Mahalo, Julie, that was fantastic. And it generated a lot of questions, this presentation, lots of questions about uses of eDNA. I know it's very cutting edge. So if you wouldn't mind just checking out what's going on in the chat there, and maybe you can go ahead and answer some of those questions, because we're going to move directly on to our next speaker. But you're getting a lot of applause, so appreciate that from everyone. But I really want to move to Andrea Blas, because she was our host this morning, and she is our next presenter. And she's the Climate Change and Invasive Species Regional Research Coordinator for the Micronesia Conservation Trust, and also one of your organizers today. Andrea, welcome back. Get your unmute, and then we'll be able to hear you. Still muted. Uh-oh.
Sorry, that was my bad. I was trying to screen share, and let's see if I can get that screen. Oh, hold on one moment. Sorry. Of course, the Zoom toolbar is right on top of where I want to click. Okay.
That always, I hope Zoom fixes that, so at some point it's a more direct.
I'm sorry. So are you able to see my presentation screen now?
Yeah, I think we are seeing the other version though. We're seeing the, yeah.
And now?
There it is, perfect.
Okay. Thank you so much. So good morning, and I think my camera is off, and just for my internet connection, I'll leave my camera off for now. But I was asked to talk about the invasive species detector dog team that we have here in Guam. There's a lot of interest from other islands that are interested for looking at CRB, and the team that we have currently in Guam is distinguished from other islands in that we're looking for CRB adults that are in outbound cargo headed for CRB-free islands. And this whole program was the brainchild of Glenn Dula when he was the invasive species coordinator for Guam, and he's now transitioned over to the University of Guam, and I've taken over as PI for the program. So a little bit of history about the program that we have. This was originally funded by OIA under the CRNR program, and it was a 3-year funding to basically create a dog team on Guam and get them trained and certified for identifying CRB with future additions of other invasive species target scents. But at the moment, what we're so that original OIA grant is ending just later this month. And so we're working on continuing operational funding from USDA APHIS and from Forest Service. They're providing us funding for another year until we're able to get this program under stable local funding from the Department of Agriculture here in Guam with the Biosecurity Division. And so there's a great legislation that was passed in Guam a few years ago that has created an invasive species inspection fee fund. This is charged to transport and cargo items that are transiting or coming to Guam. And so that fund is now up and running. And so now we're trying to get our detector dog team transitioned over to that fund so that we have stable funding. I know that Roland talked yesterday, and also Aubrey, about how expensive it is to run those dog teams and keep them operational. And so with our team, one of that big difference is that our team now is looking for the adult beetles on cargo and on vehicles that are transiting to other CRB-free islands. Whereas the previous team was looking for breeding sites of CRB out in wild environments. And so this is a big change for the detector dogs. And so at the moment, our dogs are trained to CRB scent so they can find a single CRB in cargo and warehouse and on vehicles. And we plan to expand this to other priority invasive species for incoming cargo. But also, before we get to that, we want to verify and confirm that the dogs can be trained on multiple scents. So we're also going to be looking at little fire ant, which we have here in Guam, and a lot of the other Pacific Islands don't, especially here in Micronesia. It is in Yap, but not in the other states of FSM. And so, sorry, moving on. In the last 3 years, we've been able to put together a detector dog team. So these are our local dogs that are currently working. We have we have 5 now, but originally we just adopted the 5th one, who got reassigned from the Guam Customs and Quarantine Agency. Chloe was originally an agriculture inspection dog. The other 4 dogs that we have been certified on CRB adult scents already, and they were working with handlers. And we're trying for boonie dogs, because boonies are our local term for just the jungle. But we're looking for dogs that are already acclimated to the high humidity and high temperature environment of the ports of entry here in Guam, and also the warehouses where these dogs will be working. Some of these dogs have been sourced from Hawaii, and it really takes, even though we do have a large wild dog population here in Guam, or feral population, finding service dogs or detector dogs, they need certain characteristics. And so for that, we only were able to find the one Na'i here on Guam before eventually having to source dogs from Hawaii with the Conservation Dogs of Hawaii. So they assisted us greatly in identifying the dogs. We were also able to build the semi-permanent 6-capacity kennel. So it's actually a 40-foot refrigerated aluminum shipping container that's been weighted down with a concrete floor so that this can withstand typhoons. We had Mayor come through last year, and our kennels survived unscathed. The dogs just get shut inside during the typhoon, and then and the canopies taken down, and then afterwards put back up, so that the dogs have an outdoor run. So going back to what I was talking about before, the master plan is to add additional scents for these detector dogs as they're inspecting cargo and vehicles. So we've started off with CRB. Our dogs have been certified by USDA Wildlife Services. It's actually the team that does the brown tree snake detector dog team. Those trainers have trained and certified our team for CRB. In future, we're trying to add the little fire ant scent so that can be added to the profile, because a lot of these, sorry, I'm just trying to get my pointer up. I'm not getting it. Anyway, a lot of the islands that are CRB free are also LFA free. And so if we can get these dogs doing double duty for both CRB and LFA at the same time, that's what we're trying to do. And in the future, we'd like to add the RIFA, the red imported fire ant and spotted lanternfly, to their scent profiles so that we can inspect incoming cargo as well, since we don't have those invasive species here in Guam yet, and we really want to keep them out. But just briefly, I'll go over some of the training and certification for the dogs and the handlers. So just getting CRB, they were tested in different containers at different densities. So starting off with a number, so 10, 6, 5 adults in a single container, and getting that scent, and then training the dogs to identify those scents in different containers or different cargo parts. So starting off very simple, it's that crawl, walk, run scenario where you start off just with the insect container itself and then hide it inside of a box, and then add more boxes, add more complex arrangements of boxes, and then onto pallets, and just getting the dogs. Then you can start playing with the number of CRB live specimens that are hidden to see how low of a number of CRB that they can detect once they're hidden away inside boxes. So we had the USDA Wildlife Service brown tree snake program. Their trainers came in and trained our team. They did a 9-week certification course and got trained on different items where we're expecting to do future inspections. So warehouses, pallet cargo, the air cargo for passenger baggage, and also the vehicles. Oh, my slide is not changing. The vehicles that are being shipped between islands, and the aircraft themselves. So the wheel wells, I know Ben was just talking about this. This is really important because there were a number of interceptions identified from the airplane parts itself, but at the moment our dog team is not inspecting the aircraft just because we have issues right now with getting insurance and clearance for the dogs and inspectors, or rather the handlers, to be out on the airport tarmac to do the inspections without the brown tree snake team there as well. But so our handlers and dogs went through the 9-week course and completed their certification in April of last year, and since then have been operational, doing cargo and vehicle inspections that are heading to the brown or CRB free islands. And so for the most part, our team kind of works in tandem with the brown tree snake program canine teams, so that when they go through first for the brown tree snake, and then our dogs follow through afterwards to look for CRB. And these are just very briefly some of the places where they're looking. So the cargo warehouses for the airlines, and also in what types of cargo that they're looking through, the number of vehicles or heavy equipment. And with our team, which originally was 4 dog handlers and 4 dogs, we now are down to 2 handlers, as 2 of our team members have left, but we're onboarding 2 new members actually next week, who still have to go through that 9-week training course. But with our, this is Allie here in the upper right picture with detector dog Elvis, and they actually had our first operational find of CRB on a trash truck that was set for export to Madro. And this came just actually a month before the first live find of CRB in Madro in September of 2023. So yes, since May 2023, we've had 3 live finds of CRB with our detector dogs. And for 2024, there have been no live finds, but so to make sure that the dogs are still detecting scent, even though they're not finding anything, they do a recurrent training so that our team handlers will actually bring live CRB that we have on with it from our other parts of our program. And they do the same, go back to that training cycle where they have them hidden in small containers and they hide the container of CRB in different parts of the warehouse, different parts of the vehicle, and maintain that training relationship for that scent profile for CRB. So the issue that we're having now, is that at the moment, there's no automated or centralized database for doing all of this. Right now, our dog handlers, they don't have any enforcement authority. And so all of the items that they're searching, cargo, passenger baggage, and vehicles, this is all, it's a legal limitation. They're not allowed to open it up. So if they get a positive response from the detector dog that there's a CRB scent associated with a particular item, our handlers will go ahead and do a little more searching within that item or around that item, but they're not legally able to open anything to search for the CRB. So if it just happens to be outside or on the surface, they can pull that out. And so this is just a flow chart of what's going on when they do find something. So if the dog alerts and there's no CRB found, all we can do is connect with the port supervisor and conduct a more extensive search by the port employees, because our dog handlers cannot do the search. And then also to notify the next shift of the detector dog handlers to reinspect that shipment or that flagged item, and also notify the quarantine supervisor at the destination port of this flagged shipment that should be coming in. And so I'm almost done with this presentation. So when the CRB are found, we do make it a point now to collect them and to annotate and log the data. But there's no centralized system for us to report it. And so this does get sent, the specimen does get sent off just for positive identification to the entomologist for analysis. And then, so just wrapping up right now with some of the challenges. So the enforcement and access to searchable areas is an issue. We have a small team with a large search area, and we don't have a centralized reporting system. So we're working on that. And so that's also highlighted by what Ben was just talking about. And then we'll be adding new target odors to the scent profile for our dogs. And I just want to wrap up saying thank you to our funding agencies, and then just acknowledge our team. And I've put up the contact information for myself and Garrett Certeza, who is the team lead for the detector dogs. Right. So I'm going to stop share. I know I'm just right at time, so any questions for me, please put them into the chat and I will answer them there.
Thanks, Andrea. And yes, there was some questions about the dog handling. This was the first presentation I think we've had the entire symposium, where in the middle of the presentation you saw a ton of hearts. I think that's one of the fun things about when you show pictures of a bunch of dogs, you just get a lot of hearts.
Yeah, it's that charismatic mascot, right?
Yeah, exactly. I love it. Yeah, but we're going to move right along, and please put your questions about the dog program in the chat there for Andrea. And we're going to move to our next presenter, Dr. Christopher Kitalong, who is the Vice President of the Palau Community College Cooperative Research Extension. We're so happy to have you, Chris. You want to get your camera on there? And your screen. Perfect.
I don't know if you got my share screen thing there.
Just want to hit the
Mm,
presentation
go
mode
ahead.
there.
Presentation. All right. Here, press play. The top of the screen is blocking. Hold on. There we go. Is that it?
There you go. Looks great.
I'm going to keep the same with Andrea. Connection in our region is a little slow, and at that nexus of climate change, we're just having a nice cyclone cruising through the area. So we've had some interesting winds. So I'm sure some people. So I'll just go over sort of the history. This is a kind of throw together of stuff that I've worked on with many students, and eventually with our team here at the college, and a lot of partnerships because we have many resources. Oh, just press the button. Okay. Everybody, I'm sure most people know where Palau is. As you can see, it's kind of positioned furthest west on the Pacific Islands, right above Papua New Guinea and close to the Philippines. And that'll become important later along the Kyushu Ridge, looking at the sort of on the west of the andesite line. So I'm going to just go over a quick review of what the vegetation types are, so it gives you an idea of maybe where the coconuts played important roles and how they came in and were used. So we have our strand vegetation, of course, like most of us islands with coconuts, where they come over. Limestone forests, which are, they plant some within old archaeological sites called wet, these areas near the marine lakes. Then mangrove forests, which are usually just used as a protection area and a nursery. Swamp and wetland forests, usually no coconuts. Lowland forests, the riparian forests, and the interior forest, which is our primary forest. And then there's the savanna, which we call Geth, and that's where the first components of human influence in vegetation come in. Whether there was a slash and bird community, some studies show that there were a lot of Palauans there, and that's where most of the farming took place. And then the ones that exist today, which is Misay and Mosoros, which people plant coconuts around to use in their daily lives. And of course, the farm in the Ceres, which is more modern with the tapioca introduction during the Spanish times. So this is how an old village, I don't know if you guys can hear me. Long says I'm not loud enough. But this is kind of an old village rendition. You can see the coconut palms around and how the taro patches and everything were set up. So we'll go through the coconut history in Palau because I had to go through this when I was first brought on. The pre-contact, Spanish, German, Japanese, American territory times and current. So pre-contact, obviously, we all know coconuts were spread out through all. And our ethnobotanical surveys, I can't even see my slides. Hold on 1 second. Yeah. So yeah, we have for use and not any other language. Ask some ethnographers, didn't hear anything, but still. And this is one of the Impang and copra trade with Javanese sailors from Australia through Indonesia and other areas up through to China. And Palau was kind of one of those spots along during sort of the westerly winds times, and when there was unrest within the north of Malaysia. And this is documented through money bead trades and then other metals and cloth that were seen on Palauan islanders during contact with Captain Wilson and other visitors. And during the Spanish time, copra trade was a little bit more organized, but Palau was just a peripheral colony where they picked up some of the cash crops that they brought over to Europe. And then in the German times, and this is also, we're talking with my grandmother and some other people, coconut oil became king. And as this marketing ad from old German newspaper says, just as palm trees towers above all animals, so coconut oil surpasses all animal fats in purity and quality. And this was used for cooking bread when there was no refrigeration, lasted a little bit longer. But Palauans were sort of forced, and this is encouraged to increase production of coconuts. So they were planted a little bit further up, and more intense cultivation was made. And during the Japanese times was the largest agricultural scale thing, and they're called yashirin, which we still use today to to describe these coconut plantations throughout Palau. So coconuts were important traditionally. They were used as seen throughout the islands, but it was also economically important to colonizers. So things were very well maintained and managed. So were the trees and the crops. And then we had a nice little war in Palau that took out a lot. And that was, I guess, that's Peleliu, pretty much raised and a lot of destruction. But I think the important component with respect to CRB is that there was a lot of debris that was left, and for some reason they say that it was first identified here in the 40s, and probably that debris was large. The leftover debris was the breeding source for a lot of the CRB explosion populations. And then we had the release of the virus, I guess it was 1670s to control, and then we had the CRBG. I guess we're changing nomenclature, but I'll wait for you guys on that. So as everyone said, as we saw in most of the other genetic presentations, the native populations in Southeast Asia, and they mentioned that it was first discovered, and I put quotes, discovered in the 1940s, but that's highly unlikely, as we have our own name for it, which is Ermela lius. And from talking with some of the people on the east coast of Palau, the larvae was actually consumed. So it was probably here prior, but I guess we'll have to do more genomics. And then we've had some typhoons. Guam has those, Philippines, everyone, and this is also a lot of debris. But it's also, I also kind of want to harp in that we don't really consume as much coconut, I think, on the islands as we used to. We import most of our goods, and people are buying coconut juice in a box for tourism. So obviously those were the breeding sites, this leftover debris. And then this is what the students and us started with working at, and I called it Picasso designs. When we look at the damage on the coconut trees, we have a regular Picasso art landscape in Palau. And then, of course, the impact can destroy trees. Most of these pictures over there are from Guam. And sorry, Andrea, they just had much more damage than us, so I didn't have other pictures. And we use the SPC method for qualitative damage assessments. We quantified afterwards. And then, okay, I'll go in there. I'm not an invasive specialist, and I'm a pharmacologist by trade. And so the late Yoel Miles, who was in Palau as our invasive species specialist, requested assistance on PCR work here at PCC. And we gathered a team of students to work on community-based research for the project's damage assessments, as we showed above, with no budget. So the objectives that we established during this discussion was determine the impact of CRB. Oh, sorry, and determine, okay, so we'll go through that. So we set up permanent plots in 2016 in all states using that method, and we've followed through that over the past 8 years. And the first 3 years we looked at, it was kind of strange because usually we were expecting that the damage would increase steadily, linearly, but we had this drop. So we thought there was something else going on. And as you can see, we focused a lot on the outerlying atolls because during drought time, they wouldn't have any water supply. So coconuts were their main source of hydration. So that was Kayangan, but they also dropped at the end. And then Sonsoro, where although the main village area wasn't damaged as bad, one of the outerlying islands, Puluana, ended up with 0 coconut trees at one point due to CRB. So we had to kind of act. Objective 2 was collecting samples. Well, we were very lucky to be working with Mike. And of course, we got a lot of the primer from a New Zealand group, Sean and Aubrey and Audrey and Aubrey Moore was helping. Then Mike Melzer came down and we worked with Madoka as well in Japan to get some of this stuff analyzed. So we set these assessment sites and surveying sites around the damaged sites so that we could have maybe a correlation study. And what we found was there was not really a big variation. Of course, this is with the old CO1 sequences that we're looking at, and of course we could look at them further. And the objective 3 was to determine the incidence of the virus as well. So we had CRBG and S at the time. That was nomenclature back then. This is both from 2016. And then they determined the presence of OrNV. And we found that a lot of the samples were positive. Of course, there was some conjecture that it was a contamination, but it's pretty, I guess, groundbreaking at the time. And obviously went through and kept doing it and showed some reduction and looked at some gut damage. And as you can see, there was most of them tested positives for OrNV, so there was this kind of a strange thing at the time when a G was not shown to be infected by the virus. So the summary of that work that was done up to 2018 was the question of when Guam type invaded in Palau. And again, that theory of it coming in the 40s was, I kind of would like to look a little bit further and see that as Tay's presentation showed, there was probably a native population that came up through that Indonesian, either that or during the initial colonization of Palau, and whether the tree damage here was related to G type, and we didn't show that at all. There was no correlation to that, and whether the virus was detected and the virus was not correlated specifically with non-G type based on the CO1. So the actionable items, the only thing we can do here with our very limited resources is try to solve problems. So as you saw in the beginning, the vulnerable islands were the main focus. So that island in Sonsoro was the main focus because the coconuts were being completely destroyed. So there was 0 coconuts on that fauna area, and they were starting to hit the pandanus plants as well. So what we did was obviously support physical control over there, and then worked with Madoka and Shisuke from Japan to try to see if we could get some isolates of virus that was in Palau already. And then we inoculated some of the beetles down in Sonsoro and released them. So some of the trees are coming back, and that's the same story for Kayang. When those typhoons hit, there was really bad damage afterwards because there was not a lot of green waste clearing. So we try to help get shredders and things up there to manage the green waste. And of course, some deep ground burning and things like that. So the coconut trees are kind of coming back. And we, every time we try to work with our partners, of course, we're a land grant institute. And when I came on board here at the college, I said, okay, there's a huge network. So we had the land grant people out, or I visited a couple of places and working with Mike and Guam. It's always so disjointed when we try to get different grants and funding, and everything's based on that. But what we do have in our region is this regular funding from USDA, and since APHIS and all these people are involved in allowing the release or biosecurity models for, we decided to kind of work with this multi-state hatch research project so we could officially work with US partners and try to get this on the radar within a long-term funding cycle. So we're kind of this is a planning stage, multi-state project with USDA, and we're working on a full research hatch project to kind of connect the region and make sure that we can get a lot of the academic components in. And I think that the most important is having those institutions so we can continue the research by the students, because we have this rolling every summer with the damage assessments and collections. And this is our next generation of scientists coming in. It was great to see a young person in yesterday's presentation looking at food security and the scope of that. And we hope to get more of that. Here we've had some students, and we thank Mike for taking on Nelson, who's, I guess, now working at Monsanto, but we'll try to get him back later to Palau. And then I guess the discussion now is we have to enhance these collaborative efforts. I really enjoy kind of bringing together, hearing all the stuff going on, because it was so scattered in the beginning. It was very hard for us to work, especially with very minimal resources in Palau, and trying to get the international, national partnerships and get real capacity here, but we're still working on it. And I have to bring the nexus in there. Palau can offer a site for research, nexus, between the native populations and new populations of CRB, because I think that's kind of what was shown. And I think if we get more sample sets over time, we'll find that there may be other virus or OrNV variants that we can work with. in the face of climate change, because we do have our cyclones and whatnot here in Guam and the other areas, so we can, key that in. And then mainly what I try to focus on without that research component is that it's community-based, as the young, I think it was Andrea or Andy, who mentioned making sure that the community is involved in the component, and it's not a just top-down approach and heavy outside researchers coming in, but that it's integrating these modern invasive management techniques to local practices and traditional ecological knowledge, because that'll help move things along quicker. As I showed earlier, we didn't have that problem until after the war, when the Palau's population dwindled to around 3,000. So I'm hoping that we can bring back that TEK and integrate it with our new methods. So I just want to thank you. This is the student team. That was 15 minutes, right?
That was awesome. Thank you so much. That was really interesting to hear that, having it been interwoven with that human history and how much that has influenced, the way that the coconuts have been used and not used, and, things changing, and how that might interact with the beetle. I think sometimes we have a tendency to remove humans from the equation, but we're always in there, and we have to take ourselves into account. If folks have any questions for Chris, I see a couple of questions in there. I think that it's other panelists just asking other panelists, but if you want to go ahead and respond to anything that's coming up. If anybody out there, either on YouTube or in Zoom, has any questions for any of our panelists that we've heard from this morning, please go ahead and ask those. We're keeping a very comprehensive document, so even if we don't get to these questions at the end of the day today, we are going to try and get these to our panelists, and at the very least inform the questions that our researchers are trying to answer. We do have a short break before we move to the next session, so please feel free to get up, stretch your legs. In fact, I'm going to tell you to do that. It's a really important thing to do. I'm going to turn off my camera and do that in a second, and get yourself some water and stay hydrated. And I'm going to launch a poll as well. If you already participated in the poll yesterday, not necessary to do again, but for those who weren't on the Zoom, and anybody who's on YouTube that was unable to participate yesterday, please go ahead and just tell us where you are, and share with us your experience on working with Coconut Rhinoceros Beetle. Just give us that range from never to a lot. And I'm going to go ahead and turn myself off here and take the break, and just get at that poll when you have a chance.
Hi, everybody. We are in the home stretch of this inaugural CRB Science Symposium. Thank you so much for joining us. And we're moving into our last session on day 2. Hope you had a chance to answer the survey questions. Let us know who you are, where you're coming from, which helps us to understand who is making use of these webinars. In our last session, which we'll be moving to in just a minute, we're going to have a talk and then we're going to have a question and answer session. So as Franny mentioned, please add your questions to the Q&A, to the chat in both Zoom as well as YouTube Live. And we'll do our best to get to some of those questions in our Q&A session to finish this off at the end. And with that, as soon as our next speaker is ready, feel free to jump on, turn your video on, and I can introduce you. So for our final talk, before we get to the Q&A, I want to give a welcome to Dr. Roberto Rodriguez, as well as Tim O'Sullivan. Roberto is assistant professor of Aeronautical Sciences at the University of Hawaii at Hilo. Tim O'Sullivan is with Aloha Aina Drones. And the title of this talk is Drone-Based Aerial Application for CRB. And I'll turn it over to you, Roberto, to share your screen whenever you're ready.
All right, thank you very much, Elliot. Just give me a moment to share that screen. All right, and so we've actually have kind of 2 presentations that are back to back, so I'm going to try and get through this one a little faster so that I can give my colleague a little more time. So in this one, it's basically kind of the initial experiments to develop an aerial treatment method for CRB using commercial off-the-shelf unmanned aircraft systems. So you had a couple talks yesterday regarding kind of imagery and how to process that in order for detection, so this is more on the treatment side. So it's kind of what do you get on the basic sample, and then what modifications are necessary to make it so they can perform this type of spot spray treatment. And so there are 2 main things that had to be changed in order to do this. One is to reorient the cameras, since they are designed to face forward and back and not really look down, which is what you need to look at. And then also change the nozzle configuration so that it's something that is compatible with this type of application. And so specifically, we need to meet the label requirements where for a medium or coarse droplet, and then having that swath width be less than the canopy of or less than the crown size of our coconut trees. So kind of in the box, the T10, which we have several of those kind of throughout Hawaii where different organizations is, there are 4 primary nozzle bodies, each mounted underneath the rotor. And they originally come with nozzle tips that will produce a very fine spray, which would be outside of our label specifications. So the first thing was to swap that over for a nozzle tip, which would produce those coarser droplet sizes. And then the other was moving that camera. And so we had to validate that the droplet size that we were getting out of this was correct, and our swath width was within specifications to treat the canopies. So in this plot, you can see one of the passes that we did. We were getting these ultra coarse and extremely coarse droplet sizes, and the majority of the spray was basically within 4 feet to 5 feet. So that was sufficient for the kind of application we were wanting to do. And a little bit more information on the droplet sizes. And so as far as the camera mounting, so kind of our first prototype, which was just to really get something out there in order to do these treatments, was a small 3D print in order to turn the rear-facing camera and point it straight down. And so this did work, and Tim will go through some of the results of those applications, but it wasn't quite the ideal. So it was producing these rather large, again, about a 4-foot swath width, so it would cover a large area, but it also had a dead spot underneath, directly underneath the aircraft, since all of those nozzles are at the 4 rotors. And then with that rear-facing camera, it was forcing the left and right fields to be swapped. So the pilot would basically be trying to move in one direction and the camera would be in the opposite direction. So the idea was basically switch to using the front camera and try and mount a single nozzle body rather than using the ones that already exist on the airframe. And to do this required kind of retrofitting the aircraft and cutting a lot of the tubing in order to reroute that to that single nozzle body. And so these are some of the specifications for those parts if you are interested in kind of doing this modification to an aircraft that you have, and we can provide these to anyone who's interested in those. And the other two parts are two 3D printed parts, one being to mount the camera from the front-facing portion, as well as to mount the nozzle body to the aircraft frame itself. And so kind of the process is first removing one of the styrofoam pieces that is directly in front of the nozzle pumps, which is right next to the front camera, so that you can access the wiring and then remove that camera and disconnect it to mount the new piece. And so it'll actually be mounted kind of directly between the two primary pumps. And then at this point, you are voiding the warranty that you have from the factory, but you're having to cut some of the tubing, introduce these T-brackets, which will allow you to still make use of the original nozzle sets if you need them, but also reroute tubing in order to route them to that primary nozzle you're now mounting at the base. And then as you do that, you will also have to trim some of the tubing in order to make that work. So that requires actually removing the foam guards that are around the rotors themselves in order to access where that tubing meets the actual nozzle body, and then trimming that down so that you can then secure those nozzles back. And then this is the mounting, so it's actually directly underneath one of the primary pumps using some existing holes already in the frame, so you're not having to re-drill anything. And then this is the nozzle body, so we're having the two ends coming from rerouted from those two nozzles that are at the sides of the aircraft, mounting to that single nozzle directly underneath. And then this is what that'll look like. So again, you've got a front-facing camera, and you're able to see kind of this one solid stream striking down at the actual palm. And so the benefit of this one is that it's putting all of your product right at the meristem rather than kind of scattering it all along the fronds, which is really putting products in places that it doesn't need to be. You really want to focus that in directly on that primary meristem. And so this particular project is a partnership from several other organizations. So thanks to those partners and other funding agencies. And now I will hand things over to Tim O'Sullivan.
Thanks, Roberto. I appreciate all that. Let me share my screen here. One moment, please. All right. Thanks for that one. Yes, as Roberto mentioned, I'm Tim O'Sullivan, and I'm here to present on a really exciting model for management I think we discovered earlier this year. This is basically a rapid response to an early detection of CRB in a golf course on Kihei, Maui, in partnership with MISC. So in November 2023, a single dead palm trunk yielded 17 CRB larvae that was discovered. 97 suspect palms identified within the immediate buffer area by the CRB response team. So this was a very scary thing, right? This was the first CRB discovered on Maui. A decision was made by the H2A, USDA, and MISC to treat those 97 suspect palms within a certain buffer area via drone application. To use so, as Roberto mentioned, we use a DJI Agris T10, which is modified thanks to the hard work and groundwork via Dr. Jenkins and Dr. Rodriguez. Along this was a FAA 130 exemption, which is a certificate from the FAA, which allows us to apply commercial product from the air. And basically with this drone tool set and this exemption, we are able to treat CRB populations from aircraft, aka a drone. The prescription for this area, on the right, you can see the 100 palms that were treated via drone application. Again, this was a golf course, so we basically loaded this drone up into a golf cart and drove from tree to tree along the sidewalks and treated clusters of trees as we could. The solution you can see on the left, but basically this is either a half-gallon or full-gallon drench of Demon Max per tree, which theoretically can provide protection from CRB for months. So I would argue that this drone system was an ideal tool for early detection, rapid response to a nascent population of CRB. We were able to treat over 100 palm trees exactly across the course of 4 working days. However, on average, we were able to treat 12 palms per hour. That's a palm every 5 minutes if you exclude the actual action of moving from tree to tree on the ground. A drone is ideal for this kind of work, I would argue, because the mobility allows you to treat trees that you couldn't access via helicopter, crane, ladder, that kind of thing, especially when they're oddly shaped. You can see this coconut tree here on the left is kind of in the back in a bunch of chiave, so it would be quite hard to get up there via human or crane truck. So we argue drones are ideal for that. More than anything, it is a huge safety tool, right? Because for a human technician to climb that tree and apply said pesticide, they themselves are being exposed to the pesticide, as well as risking their life by going up far above the ground. Furthermore, a drone drench, as Dr. Rodriguez pointed out, can be very, very surgically applied to a tree, mitigating for wind factors and such. And more so, given that these systems are equipped with high-end data collection software, we feel quite safe that the amount of pesticide applied to the tree was the correct amount. And after this action, it's been some fantastic news. To this date, we have not seen any other CRB on Maui since initial inspection. So in a lot of ways, we would argue this technique was a success in at least controlling for CRB in the immediate buffer zone of the initial detected population. So if you excuse me for a second, I'd like to offer my own company. So we are Lohana Drones. We are a Part 107, 137 certified company. So that allows us to apply aerial product from pesticide or commercial product from the air. We also hold air applicator license from the HGOA. Together amongst the team, we have 30 years of combined experience using drones for conservation efforts. And we are currently the only drone company in Hawaii I'm aware of that has direct experience treating CRB. Here is my new family member. It's a DJI Agris T20 as opposed to T10, so it has twice the capacity of that initial T10. We've modified the system, as Dr. Rodriguez has pointed out, for a number of different spot treatments. We can fly for a long time, and we have these advanced safety operations. And finally, most importantly, we are a conservation-based agency, and our pricing really, really shows that. We're here to save Hawaii, not to make a living, as my family members hate to say here. So we would be very, very interested in helping protect Hawaii from CRB and frankly, across the Pacific for years to come here. Again, this model of rapid response to early detection of CRB population via drone application, I think is a fantastic model that we should consider and think about as time goes on, given both the efficacy, efficiency, safety, and the cost reasons. So if you have any interest in working with us, you can see my contact information here. Again, I really appreciate you allowing me to solicit some business during a fantastic symposium with a lot of smart people doing a lot of smart things. At that point, I would say thank you to my co-authors, and thank you all for hosting this amazing symposium. Aloha.
We do have time for a couple of questions. Thank you so much. So feel free for both of you to leave your cameras on if you have time. So first one I'm seeing in the Zoom chat. So the initial detection was 97 trees at the golf course, and those were confirmed to have CRB.
I'm sorry if I was confusing there. So a single tree was found to contain 17 CRB larvae. And based off of some GIS work, we basically created a buffer around that initial detection tree. And so there's 97 trees that were treated were suspect trees, even though they showed signs of damage, or more so they were in the immediate immigration area of a CRB specimen. So only one tree showed CRB, but we treated 97 proactively to protect them and hopefully control if there were CRB individuals present within those trees.
Great, thank you. There's another question here. Is emergency drone treatment being conducted on Kauai? And then I'll add to that other places that you're working.
Yes, well, I think Dan Jenkins, if he's on, might want to jump in here, or Roberto, feel free to hop in. I know that Dan Jenkins cut his teeth in his initial drone work on Kauai and Oahu. However, I'm currently not familiar with any going on right now in Kauai. If someone else in the room is, I'd be very curious to hear. And if not, we'd be very interested in helping.
And we've got another question. Maybe we can do one or two more. So please keep the questions coming. So any indication of the cost of using drones plus pesticide per tree in Hawaii?
That's a fantastic question. Gosh, so we are currently, I guess I'm soliciting some business here, we are currently charging $175 per hour to spray via our drone platform. As I said, we average about 12 trees per hour, so you can break that down there. But I would argue maybe $20 per tree, if not less.
Thanks for that. And there's a comment. This is great news, possibly an eradication, only the second CRB ever as far as I know. Yay.
Agreed.
So keep our fingers crossed, but we definitely need to have more than one eradication success story in the Pacific. So hopefully this is a pathway to get there. I'm just going to check the CRB live YouTube to see if there are any other questions there. And I am not seeing any at the moment. So thank you so much.
Thank you. Hello, everyone.
All right, well, we're the final home stretch here. We're going to be moving into our Q&A session. So we have some questions prepared, and I'm going to introduce our two moderators for the Q&A session and then hand it over to them to take it away and finish us off here. And 1 second, just pull up my document, which of course just disappeared because that's the way it works. And so moderating our session, our Q&A session here today, we have Chelsea Arnott, the Invasive Species Coordinator with the Hawaii Invasive Species Council and the Department of Land and Natural Resources, Division of Forestry and Wildlife, and Christy Martin, who is the Program Manager for the Coordinating Group on Alien Pest Species at the University of Hawaii at Manoa. Thank you so much, and I'll hand it over to you both.
Yeah, thanks, Elliot. Appreciate it. And rest of the planning team, this symposium has been great. I've learned a ton, and this is always my favorite part, is having discussion and time for Q&A. So hoping we can get through a few questions here, but I know the planning team is, if there are unanswered questions, like Franny mentioned, they are planning to try to get responses to those questions and put them together with a summary at the end of the symposium that will be sent out to folks. Great.
So we haven't practiced at all, but if you would like, we can take turns on questions.
You're going to see how much we haven't practiced right now. But maybe first, if we could ask the panelists that are still here with us, we have some folks from yesterday's symposium and then the folks that are still able to join from today's. If you could turn your cameras on, that would be wonderful. And we're going to kind of round robin through some of the questions that came through the chat over the last 2 days. Some of the questions, they're bigger, broader questions. And then we do have a few questions that are more focused to those panelists, and we'll make sure to direct those. But if any folks, any panelists on here have thoughts on a question that might not have been directed to you, feel free to chime in. But Christy and I will be moderating, so we'll try to keep things concise so we can move on to other questions that are here, because we got a lot. But yeah, Christy, maybe I'll shoot it over to you if you want to start with the first question.
Sure. Thanks so much, Chelsea. Thanks, everybody, for great presentations. I learned a ton. I've got lots of notes now. I think a lot of questions really focused on pesticides. So it's kind of a two-part question, I think maybe starting with Keith. First of all, what are the best alternatives to pesticide that are available right now? People are in need. And then I guess, we're going to talk a little bit after that about what are the best ways to minimize impacts from pesticides when we absolutely have to use them, which we do. So, Keith, are you able to answer that first part? Best alternatives available right now?
Yeah, and it depends where you are, because I know we have some people from across the Pacific, and so we heard a lot about biocontrols, and most of those are not available currently in Hawaii, but there is some work going in that direction to get those here in Hawaii. But green waste management is probably the number one. As you saw in my talk and other people's talks, it can be very effective as long as it can be done on a landscape scale. That's the biggest thing. And then There's also use of netting in specific ways on breeding sites, potential breeding sites, and in trees is another alternative. So, but sanitation by far is the most effective. Even if you're using pesticides, sanitation is strongly advised. So, controlling all the potential breeding material at a landscape scale.
Thank you. Turn it over to Chelsea.
Thanks, Christy. And like we said, if other panelists have anything to add on to these questions, feel free to just unmute yourself and add on. I'm going to go for one of these bigger questions that might circle around to a few folks here. How can we support communities where eradication is no longer an option? A lot of what we have seen over the last 2 days comes from, a lot of efforts that are being taken on by the communities because they don't feel supported here in Hawaii with the actions and the tools that we currently have. So maybe we can hear from a few folks from the other Pacific Islands and have some lessons learned there. Chris, maybe, yeah, you would be great to kick us off.
Sorry, can you repeat that? I was talking to a student.
So the question is, how can we support communities where eradication is no longer an option?
So just the stuff we've done is I think the best example would be like when French Polynesia eradicated the Orientalis. It's community-based messaging and having people from the community that are not prominent, but like, because sometimes prominent members are politicians. So it's just having community action committees put together, local people having it in their own language, and then kind of having them look at it as an economic deterrent. I kind of stopped a lot of the work here because people weren't seeing major damage. But what people brought up was like, okay, now they could damage the beetle nut trees, and then everybody got crazy. So it's finding those components, and that's where the TEK, where the working directly with the communities and the students to handle it, it makes things less expensive and stuff that lasts longer. So, and what we've just been trying to do is make sure that we all talk together and share. We're trying to get the virus out. If you got it here, if you got new methods there, just share as opposed to this grant driven kind of drive, have like a goal and make sure that the community is there with you. And then I think if it's just grant driven stuff kind of stops. That's what I've seen. I'm not professional in invasives, but that's what I've kind of seen. If there's no sort of specific granting mechanism, it goes away right after. But when it's a community driven thing, they kind of put activities aside for members of the community that are already sort of funded. And when it goes to the universities, and like this is why I came to the land grant system, is that there's a set structure there and people there for longer periods of time, and you have a rotation of students coming through. So I really see the universities and high schools and even down to the Head Start programs, which we're working in now, as really peer mentoring with that traditional knowledge component and the Western science coming in together. So it's not separated. It's under one integrated model. And they like integrated pest management. I said integrated TEK and pest management.
That's great. Thanks so much, Chris. I see, Aubrey, you had your hand up, and then Andrea, you want to add on?
Yeah. So one of the talks yesterday mentioned a great idea, and that was having community trapping. And the idea was to put up panel traps, pheromone traps, and have people monitor those in their backyards. I think a better way would be to use the Tekken idea, the netting. You can use that as a very cheap and efficient pheromone trap. All you have to do is attach it to a fence or even wrap it around a coconut stem and hang a lure there. These lures aren't cheap, by the way. They're $4 a piece, and they only last about 10 weeks. But the netting is extremely cheap. And here's the kicker. You don't even need the lure. If you have a small pile of green waste, you just cover it with a piece of this Tekken, maybe 4 square meters of it, and the beetles will very quickly start arriving. And once you get a bunch of males in there, they're your lures. They're putting out the natural Arictolure. So this netting is available in bulk. It comes in bales, which are 17 feet wide and 1 kilometer long when it's stretched out. It's about $800 for one of these bales. So if you had a bale on each of the Hawaiian islands and just cut it up into squares and gave that away, that would be a great way of starting things off, because you're actually doing sanitation at the same time, right?
Yeah, mahalo, Aubrey. I think you just covered 3 questions we had there. So
these piles can be used as early detection. You can have them in places where you don't expect to find rhino beetle, and they will likely be discovered first as adults on those piles.
Right on. Thanks, Aubrey. Andrea?
Whoops, sorry, I was on mute. So I think Aubrey covered some of what I wanted to say, but yeah, and Chris as well. So just the community outreach and education is one of the best ways that we can do this for those communities where eradication is no longer possible. But one of the other things that we're working on is really working with landscape companies and also with the military contractors to integrate the green waste management and requirements for hot composting of the green waste afterwards. And one of the, so we don't have there's only one facility on Guam that's licensed for the hot composting, but all of the military green waste is required to go there. But actually making that available to the public is something that would be very helpful in just getting those hot composting facilities available for residents to bring in their green waste so that they're not creating more breeding habitat for CRB would be very helpful. Another thing that our division chief for biosecurity has been working on with the emergency response teams here in Guam is getting the hot composting and green waste management integrated into our disaster response plans. So after Typhoon Mawar came through last year, that was a big issue, is that the green waste collection sites were placed right next to the port, which is the high pressure area or risk area for escape of CRB from Guam to other islands. And so figuring out with the emergency response teams in each island, where are those green waste collection points going to be located and situated after a natural disaster is really helpful. And so that's something that our Department of Agriculture is working on now.
That's so great. So many great solutions and lessons that we can bring over here. And I'm definitely seeing some of that already happening here on Oahu. I think we should move on to the next question. But again, panelists and even folks attending, if you have thoughts or ideas on these questions, just put them in the chat so it can be recorded. Christy, I'll shoot it back over to you.
All right. Would you, I'm going to skip to what is the recipe, if we know it yet, for an eradication? I know small population, early detection, but we've had so many learning points along the way. What's going to be the recipe for the next early detection? Or maybe it's going to be on the Big Island. Anybody want to take that one?
This is Aubrey. Yeah, I'd like to address that. So I was really surprised about this, learning that Maui has a potential eradication that's happened this year. That's fantastic, because the only other one that I know of about was on a very small island belonging to Tonga called Keppel Island, and that was done way back in the 1930s. There have been many, many eradication attempts since then. I mean, if your island is invaded by rhino beetle, you've got to attempt eradication for sure. But it's not impossible, but highly improbable with the tactics that we have currently available. I'd like to know if anybody knows what's happening on Majuro. You know, Majuro got hit by rhino beetle last year. Now, Majuro is about half the size of Keppel Island. There's a real opportunity there to eradicate, to learn something about eradication. It's only a single flight from Honolulu to Majuro. That could be a really good, interesting testing ground. Is there anybody here that knows what's happening on Majuro? I've kind of lost track of what their success rate is or what's happening.
I actually just had some pictures shared on WhatsApp from the team in Majuro yesterday. So they're doing periodic surveys and searches again for breeding sites so in Majuro, the initial find was there was a collection of a CRB adult found on the ground at the airport in Majuro, just like 20 feet from a CRB panel trap. So it wasn't in the trap, it was just on the ground. And then the very next day they went out and they found the breeding site, which was about a mile down the road from the airport. And since then they've had a response team that goes out and does periodic searches. So every breeding site that they can find, they destroy. Yesterday, Kiavin was sharing images of their catch from a breeding site they found and destroyed yesterday. So they have another survey. Right now, the CRB and Majuro are still concentrated in that 1 to 1.5 mile radius from or actually west of the airport. It hasn't made it to the east side of the airport yet. And so it's still concentrated in that area. And so they are still doing periodic searches or regular searches for the breeding sites. So they are making a dent and they're keeping it contained.
Thanks, Andrea. That's good news that they are making some progress. Do you know who's funding that project?
I'm not sure with who all of the funders are. They have some emergency funding from the RMI government, but they also have support from FAO, from United Nations, and then also from US Forest Service and a few other partners. So I know like myself and then your technician from Guam have gone out there for technical assistance, as well as some of the team from Hawaii have gone out. Mike Melzer was there and Ryan. So I'm not sure. And then also experts from India funded by FAO were there to support. And I believe also Mark Arrow, who's unfortunately left, couldn't stay for this panel. I think he also was there for technical assistance.
Yep. I'll just chime in. I was there for the micro games, and prior to that, our other land grant guys are helping out on the ground with extension with their Bureau of Agriculture. And they initially got the traps out from Rollin. We just had a call with them to set that buffer area. And it's interesting because they have set winds. So after talking with some of the fishermen, they started setting up the traps in areas where the winds are going to take them. So it was a very interesting community component too.
Do you know if anybody's checked the islands that are on the north side of the atoll?
Yes, we set up ground traps out there with pheromone lures, and within the quarantine ministry or the quarantine division, they're making monthly checks.
Sounds like a lot going on. Thanks so much. That's a really good conversation. I hadn't heard any of those updates from Majuro, so thanks to everybody for for lending expertise and knowledge on that. Yep, go ahead, Chris.
I just wanted to ask Aubrey, could you explain more of what happened on that small island and how they eradicated the beetle?
Yeah, it's a small outer island of Tonga called Keppel Island. That's not its real name. I can't pronounce the real name. And it was about twice the size of Rota. No, smaller than Rota. It was smaller than Rota.
I think you said 16 square kilometers.
Yeah, 16 square kilometers, which is a tiny island. So they got infested, and there was an old German guy living there, and he says, I'm going to eradicate this. And they knew about sanitation, you know, they knew about the grubs and the adults. So he got permission from the chief to use as much manpower as he needed. So for a 7-year period, they scouted the whole island, found all the breeding sites, destroyed them when they found them, and the beetle went away. It was eradicated. Took a lot of effort and a long time. And that's a tiny, tiny island. Surely with our knowledge and tools that we have available these days, we can do a better job. But I think you're going to be
But hopefully we
luckier
can bring the
on
QC.
smaller islands like Majuro, for sure. And then maybe Rota. Rota is a good place to use as a test bed as well.
Yeah,
I think that also key that the cultural leader was involved. So he got the community initiated. And with the technology, if we can bring the community and the students and fuse that into one sort of, I want to call it education because it has all components. That's what I was mentioning before. Integrate those approaches because that is effective. And now we've seen Maui with one effective. So if we can fuse those things, I think it'll be working across the islands.
Yeah, this is great. I do want to move on to the next question, but yeah, Timo, I see your hand up.
Just really quickly, you know, I am biased here, but Aubrey and the rest of the team, I'm curious what you guys think. I've been to Marjoro. There's a lot of coconut trees, but not too many. What if we treated every single tree in Marjoro via drone? Would we feel like that might? What would
Sure.
that
Has
be
anybody
for eradication
recipe? Has anybody estimated the cost of that? It probably wouldn't be that much. I mean, you just fly along the island. It's about 30 miles long, but it's just like the width of a road, basically.
Yes.
Speaking to some of the regulators over there, I don't think that pesticides are an option that will fly with either the regulators or the populace. And I think that's going to be the biggest barrier.
And Keith, may I ask why that is? Is it because of the crop production and the agroforestry culturally? I'm curious.
I think it's just pesticide hesitancy, probably for a number of reasons, human health and ecological concerns, probably.
Yeah, I'm really a proponent, I think, for the idea of evergreen, given that it's a product, you know, used for organic food production. It's still a pesticide in name, but in action, it may be something different. Yeah. And Dr. Jenkins has found some fantastic usage for that. So,
evergreen is a pyrethrin. It's a pyrethrin, and then the cypermethrin is a pyrethroid. It's an artificial pyrethrin. And by the way, this cypermethrin is not a very strong pesticide. It's the same stuff that they put in Raid that you spray in your kitchen. It's got a very, very low mammalian toxicity, but I know a lot of people don't like using pesticides at all.
It's just getting community buy-in.
Yeah, the pyrethrin you can actually use in a food processing plant.
Well, this is great. Yeah, this is touching on some of the concerns people have expressed with, you know, edible foods and spraying products on that. So I think there's some great case studies in here. I don't know if they've all been compiled together, but just bringing these minds together into one session, I just see like all kinds of great products that can come from these conversations. I do want to move on to, we have a lot of questions and we're definitely not going to get to them all. But one question that came up into the chat, and I know this is kind of the other hand of it, you know, does it take for eradication, and what do we do when there's, you know, CRB is widespread and established in these islands and it's circled back to biocontrol. So specifically with Hawaii, what is the barrier to more research on the fungal and viral biocontrol, or even other classical biocontrols? And Keith, I think you were kind of answering some of that, but it'd be nice just to have a concise response. And if other folks want to assist with answering that, that would be great.
Yeah, in Hawaii, there's a lot of interest, and I think almost everybody wants the biocontrols. I think the biggest barrier is that there's legitimate fears that there could be unintended consequences. So some safety testing is appropriate and legally required. And even the containment for the testing is a difficulty. So having a facility that's suitable, there's not a facility that regulators are accepting as adequate containment. But there are efforts underway now to get both short-term and long-term increasing capacity for that containment and testing for biocontrol. So hopefully that will happen soon, but we don't have a date on when that'll happen. But everybody's interested and thinks that particularly the nudibvirus will hopefully be a real game changer for CRB. It's just a matter of time and effort.
Thanks, Keith. Christy?
Yeah, so there's
go ahead, Aubrey.
So I've worked on biocontrol of rhino beetle for the last 15 years. First of all, we introduced the Metarrhizium majus into Guam, and we use it as a classical biocontrol agent, and it spread very rapidly. throughout the island, and it's still there. It has some effect on the population, but not that much. But historically, the virus has been the sustainable, long-lasting relief from rhino beetle damage. You know, for 30 years, there was no spread of the rhino beetle because the populations on new islands was being kept down by introductions of the virus. So the advantage of using biological control agents is that they reproduce. So you put it into the population once, and if you're really lucky, if you have a good candidate, it will reproduce and spread by itself and maintain itself in the population, sometimes forever. It kind of reinstates the balance of nature. And the nice thing about biocontrol agents is many of them are species specific or close to being species specific. So there is no non-target effect. And with rhino beetle virus, with OrNV, human populations have lived in the presence of this virus for eons. If you're living in a native coconut environment, there's probably OrNV there because almost all of the populations in the native areas have a high load of OrNV. So even though we failed to find an OrNV which would help us control the beetle on Guam, I haven't given up on that. I think it's just a matter of matching the right isolate to the beetle that's on Guam. So that's my take on it. I think OrNV is going to be the long-term sustainable solution.
I want to ask a question, sort of a combination of several questions that have come up in the chat. And that is, maybe this is targeted a little bit towards Matt. You know, you talked about the challenges of finding those chemical lures to make a more efficient mousetrap, CRB trap. So are there things you still want to try? And I guess another question was that have you tried looking at the opposite? Have you tried looking at repellents, or has anybody?
Yeah, so I can't speak authoritatively on whether what other people have tried and isn't in the literature, but at this point I don't know of people trying things that are repellents, and I think that they tend to be a little harder to use. Yeah, I mean, folks picked up on the thing, and we had multiple people talk about the fact that compost seems to be so much more attractive, and that's actually from the bioassays that we conducted, that seems to be the case. I think my concern there is how much different is it from the background, and if you've got lots and lots of coconut trees, lots and lots of compost out there. Can you use that? Do I think we can make a better mousetrap? Yeah, sure. I think we can. And yeah, I think probably looking at compost, looking at some of the coconut associated volatiles are something that would be a possibility. Like I said, I tried to stay away from the chemistry a little bit, but this molecule has two different forms. So the Eryctolore, and we're putting both of them out there at the moment. So a left-handed and a right-handed version. And there's really not been a lot done to see, does the beetle care about one versus the other? And there are a lot of insects that do. They like one and they don't like the other. And so there are some possibilities there. So some things that are possible. I'm not sure that it is going to be the silver bullet. I don't think that this is something that we can trap out. And I know I'm influenced by Aubrey on that. I think a lot of what we can do to make this better are the traps and the lights. Those are things that I think are probably lower hanging fruit. But as a chemist, I'd love to get in there and see if there are some things that we could do or continue to do the work and see if we can improve the lore.
Yeah, mahalo.
Is there a day 3 just for discussion? Because we're already at our time right now. Yeah.
The problem with chemical control is you got to disperse the material. You got to get it out there. And that's a big problem with this pest because on many of the islands, it's not an agricultural pest primarily. It's not found just in plantations. It's found throughout the environment. So you got to cover very large areas, basically the whole island. Like in the case of Guam, there's coconuts all over the whole island. You can't, they used to spray the whole island. They did it for malaria and dengue after the Second World War. You can't do that anymore. But what you can do is you can put out these little beetles that have had a dose of virus, and you just let them fly around, and pretty soon they'll cover the whole island, spread virus throughout the whole island, and you got sustainable biocontrol going on. And to some extent, that's what's happened with the metarhizium. Many of the breeding sites that we visit, there's already metarhizium there. And prior to us getting the beetle, we didn't see any metarhizium mages on the island. We actually brought it in from the Philippines and released it.
Thanks, Aubrey. We're so lucky to have you. still how much you love your work and still participating. So feel very lucky to have you in these conversations. This was a great conversation. Obviously, we need a whole nother day. I'm going to push it back over to Franny and the planning team because I think we're pretty much at the end to close it out.
Thanks, Chelsea. I really enjoyed this part. I mean, all of it was so good, but really hearing this back and forth. And it really feels to me, although I don't want to do this to us, Elliot, Keith, Andrea, but I don't think this conversation is over. I think there might be a future for us to bring folks back together again. But it's just such great information. And I really want to thank all of our presenters because you had so much to share, not just in your presentations, but in this session here. And I just feel, again, there's just more knowledge, I think, that is out there that we really could gain so much with future conversations on this. So thank you all. I want to thank all of the folks who attended. I know it was really tough to sit in front of your computer for a few hours and try to engage with all this information, but I can tell by the questions that have been coming in that people have been engaging with this information at a really high level. We have recorded all of these questions. If your question did not get answered, it is not bye-bye, so what? We're going to be putting together a communications document, and we will be sharing that with the panelists and following up, and then we will share some information out along with the full recording of both days, probably next week, to everyone who is registered, so that everyone will have an opportunity to hear these presentations again, go back, check on anything you missed, because I know I have to do that, and also following up with that communications document, which will capture a lot of these questions and a lot of the answers that we get from our panelists. So once again, just really appreciate that. Team, anybody else have anything they want to add before we say goodbye for this year?
Randy, you folks broadcast this to the YouTube channel and it will be available there.
Yeah, and yesterday was a little snipped off at the beginning, but we will share out the Zoom that has Aubrey's full presentation from yesterday. Yeah.
A huge thank you to everybody. This has been amazing and definitely want to continue the dialogue. So thanks again to everybody.
Okay. Mahalo.
Aloha. Nice to see you again, Aubrey. Catch you in PI.